गुड मॉर्निंग बच्चों, गुड मॉर्निंग. In one or two classes, some glitches there. They are doing it. Already gone. Very good morning, everyone. All good. Seeing your struggle, I remember my days. In fact, every examination, not only the NEET, every examination after passing through NEET, then the life is putting a lot of challenges. Yeah. Good morning, bachcho. Good morning. Can you hear me? Very good morning. So meanwhile, I will tell you the plan now how we are doing, and in case if you have anything to share, you can also share it. So what the plan is, roughly the session is for 5 hours. And like 50 questions is geology questions. So on an average, 10 questions per hour. So we'll be preparing for 10 questions. Of course, we'll be doing more than that. From that, we are expecting 10 questions. Sometimes 9, 11, that may happen, right? Roughly 10 questions every hour. So we'll divide like this. In case if the class is going more than that, suppose we decide, let us say we decide to start the second year. The second year is reproduction. Reproduction, then reproductive health. After that, we have those the genetics chapter, right? So it means reproduction, 4 questions, reproductive health, 3 questions. The 4 and 3, 7, right? Now the first chapter of genetics, that is physical basis of inheritance, wherein principles of inheritance, wherein we have those genetic disorders and everything, mutation, pedigree, that is 2 roughly, and the last one is the Human Genome Project and DNA fingerprinting, right? After that, again 2, so it become how many? So 4 and 7, 11. So roughly this will be the plan. Now we are starting it at 7:57, right? So by 8:57, we have to be almost at the Human Genome Project, or at the end of that, in fact. Getting my point? Getting my point, everyone? Getting my point? So I don't know what this TV means and what this TV means. Let him come, then I will ask him whether the YouTube people are there or our own college people are writing these. So in the first hour, we will be doing up to and from animal reproduction up to Human Genome Project. This makes our 10 questions for the first hour. So like that, I know how we have divided the whole and how they do that roughly. So 3-4 questions in human reproduction. Now this chapter is quite easy because most of the things are already in the mind. There are very few things which people can just forget. But still, a recall is always good. So where is this? MSD. MSD is, this one is human reproduction and reproductive health. Okay, so we have cut the topics from the NCERT book. Now what is required is you please open your NCERT book. Suppose if I am doing this, male reproductive, and in case if you think that you have just now, a few days back you have done that, so meanwhile you can open the NCERT and you can later on the topics, for that you can keep on preparing. So it is a little different from the regular classes that we have, wherein you have to be in the class as the pace is going. ठीक है बेटा? तो suppose if, like for example, I'll tell you how it will. So what are the primary sex organs? The primary sex organs are the organs that make gametes. Okay, and what are the other, all other reproductive organs are called secondary. So primary, just remember, primary sex organ is called gonads. It's called gonads, right? Male gonads are testes, a pair of testes, and female gonads are a pair of ovaries. Make sense? Very good. Now, yeah, we got that. Testes, let us see the dimensions of testes. Is everything going good? I did not, because he has not come. Board is not clear. Can we send this file to your classes? Let him come. I will ask him to send these files to the classes. Hello? No, beta. क्यों आ रहा है बार बार? If in any class, if some issues are there, screen, okay, what I can do, I can ask him to send these files to your classes so that, you can have them. And meanwhile, you can just listen to the voice. I'll be giving the voiceover and making you learn that in the classes, this file can open, right? Let him come, beta, ये कर देंगे अभी, ठीक है? AC अभी थोड़ा चल नहीं रहा ढंग से. That's why I'm starting. No worries, within few minutes. Meanwhile, I'll share a very lovely quote. If I can have that. We all have breakfast in the morning. Almost all of us, you too, me too. Having food on the table is not something that is, we can take it for granted. Now, study says that woke up in the morning with more health than illness. Little illness, headache or backache is very little, but you can get up, you can brush, you can go to washroom, you can take bath, right? So if you woke up in this morning with more health than illness, you are more blessed than the millions who will not survive this week. Even today, right now, many people are dying right now. So we are much blessed than them. If you have never experienced the danger of battle, battle means the country is into war. Battle, the loneliness of the imprisoned there, the agony of the torture, or the pangs of starvation. Pangs is a word used for when the stomach is empty. So the type of sensation that we get in the stomach, right, that is called hunger pangs. So pangs in the stomach, you are better off than 500 million people in the world. 500 million people. The country is normal, politics is going normal, there is no battle, there is nothing. No bomb, like, being bombarded on our heads, just like what has happened in Gaza, just like what has happened in Ukraine, right? So we are much better. If you can attend a classroom or not attend one without fear of harassment, arrest, torture, or death, you are more blessed than 3 billion people in the world. If you have food on the table, clothes on your back, a roof overhead, a place to sleep, you are richer than 75% of this world. 75%. So we are in the top 25%. Only for these Now, lastly, it says that if you have money in the bank or in your wallet or spare change in a dish someplace, you rank among the top 8% of the world's wealthy people. If you can read this book, you can listen to my voice, you are more blessed than over 2 billion people in the world who cannot read or who they cannot understand. They can hear, but they can't understand what is being spoken. So you are better than 2 billion people in the world who cannot read or cannot listen. So count your blessings and remind everyone else how blessed we all are. So let us count our blessings. So a lot of things are, we are much blessed, we are much blessed, right? Throughout the session, our health is not giving us too many challenges. We are much blessed. Okay, bacho, I think if any issues are there, by now they would have been resolved. So let us see the testis dimension. You know that 4, 5, 2, 3, 4, 5, right? So like, how do we do that? 2 to 3 centimeter thick, 4 to 5 centimeter, the width, and length is 5 to 7. Okay, so like that, the dimensions of the testes lie inside the scrotum in the human beings, in the mammals, so that spermatogenesis can take place. The temperature, do you know the temperature? How much temperature lesser in the scrotum than inside the core of the abdomen, right? Anyone can write so that I can understand that you people are also listening to the classes and also understanding which chapter we are doing, right? So testes pair oval in shape, 4, 5, 2, 3, 4, 5, 2 to 3, 4 to 5, 2 to 3, 4 to 5, located outside the abdominal cavity in a pouch called scrotum, and the temperature is how much? 2 to 2.5 centimeter. 2 to 2.5 centimeter lesser than the normal body temperature. So this comes out to be 34.5 to 35 degrees Celsius. How much? 34.5 to 35 degrees Celsius. Covered by dense connective tissue, consists of 200 compartments called testicular lobules. So in one testis, 200 compartments. Each testicular lobule has 1 to 3 highly coiled seminiferous tubules. And the function, what is happening in the seminiferous tubule is the spermatogenesis or sperm formation. Two types of cells inside the seminiferous tubule, the male germ cells, and the other one is the Sertoli cells or the nurse cells, right? And the function of the testis in whole is spermatogenesis, and the second one is a hormonal. It secretes the hormone testosterone. Now this is the diagram. I think you can identify all of them, which is the unpaired gland, accessory sex gland. The unpaired accessory sex gland is the prostate gland, whereas the seminal vesicles and the bulbourethral, these are the paired one. Makes sense? Now the female reproductive system, let's move down to the female. These are the two types of cells, and they are called the nurse cells because they provide nourishment to the germ cells, to the spermatogonia. Okay, so now let us come to the female reproductive. The female gonads are ovaries, they are also paired, and now the only length is given 2 to 4 centimeter in length. If we compare this to the testes, how much is that? 2, 3, 4, 5, 2 to 3 and 4 to 5, 2 to 3, 4 to 5. Only width and the length is given. Connected to pelvic wall and uterus by many ligaments, right? Covered by thin epithelium which enclose ovarian stroma. Stroma divided into two zones, peripheral cortex, inner medulla. In the cortex, there are so many follicles which are, in various stages of development after the puberty. Function of the ovary is to produce the egg, and another one is the sex hormone, right? Now, how many sex hormones in general? You can just remember 3 of them, and these are: number 1 is the estrogen, number 2 is the progesterone, and anyone can write the third one, which the ovaries produce. The third hormone. Anyone remember the third hormone? I'll give you a hint. The cells of corpus luteum. The cells of corpus luteum, they are producing. Hint number 2. Normally the secretion is considerable in the end stages of pregnancy. In the end stages of pregnancy, what is that? Very good. It is relaxin. So please remember that relaxin is also produced from the ovaries, not only by the placenta, also from the ovaries. Many people are writing, I'm really very much, I thought that still in some class glitch is going on, and okay, so people are writing, and this is really very good. Okay, commendable. So a question came like, this hormone relaxin is produced in the ovary by, and simple answer was the corpus luteum, right? Achha, one more thing, please understand that the corpus luteum survived throughout the pregnancy. Now, some people think that the corpus luteum dies, okay, or degenerate after 3 months of pregnancy, after the first trimester. No, the reality is after the first trimester, there is another endocrine structure which is formed, and you know the name of that, that is called placenta. That is after the first 3 months, right? So during the first 3 months, the only source of progesterone is the corpus luteum. After that, corpus luteum along with placenta, right? So both are doing. Please remember that to maintain corpus luteum, what is required is HCG, human chorionic gonadotropin. And you know that placenta also produce human chorionic gonadotropin, HCG. Now, if the placenta produce that, that means the corpus luteum is surviving throughout the pregnancy. Please understand that. Right? Okay, very good. The uterus, we can divide the uterus into 3 layers: endometrium, myometrium, the perimetrium. The myometrium is the thickest one, and it's made up of smooth muscles. The endometrium, it is the inner glandular layer. It lines the uterine cavity and undergoes cyclical changes, right? Please remember or revise the length of the fallopian tube, 10 to 12 centimeter long. Okay, and the various part of the fallopian tube is starting from the ovaries. So we have the infundibulum. Okay, infundibulum has many fimbriae. The shape of the infundibulum is funnel shaped. After that, there is a wider part called ampulla, and the narrower part called isthmus. Remember, ampulla is the wider part and isthmus is the narrow part. So obviously, the site of fertilization is ampulla. Please don't misunderstand that. Ampulosthemic junction. Old NCRT has written this mistake, ampulosthemic junction. Medical book says ampulla, and the new NCRT also says ampulla. So please, in one place still, in the current textbook also they have retained that ampulosthemic junction. So please understand, it is only the ampulla you have to answer, right? Okay, so external genitalia, mammary gland. Breast comprised of mammary lobes. Each mammary lobe has got one duct, and that duct will join to form the ampulla, and ampulla will open at the nipple by the lactiferous ducts, right? So how many lobules are there in each breast? 15 to 20 lobes or lobules, right? 15 to 20, these are called the 15 to 20 lobules. Now each lobule has many these we can say these alveoli. Here also we can call them alveoli. Now please understand that the female sex hormone, we can write now, we can highlight, okay. The female sex hormone, which is estrogen, is mainly helping in the development of the ducts, and the female sex hormone progesterone is mainly helping in the development of these alveoli. Now these alveoli open into mammary tubule, and then mammary tubule open into mammary duct. Many mammary duct join to form mammary ampulla, and ampulla open at the nipples by the lactiferous duct. So please remember the order. Okay, the function of this is, lactation, spermatogenesis, and oogenesis. Now this place, I think some people have still doubt about spermatogenesis and oogenesis. Please remember that the story begins with, can we open the blackboard? Teacher cannot remain silent without writing. Right. So the gametogenesis, whether in case of male or in female, so gametogenesis, it requires 3 steps, and I call them MGM. So MGM, the first M for mitosis, that is, multiplication. Multiplication occurs by mitosis. Then growth. So one of them undergo growth, and finally this M for maturation. Maturation occurs by meiosis. Maturation occurs by meiosis, and multiplication occurs by mitosis. Initially, mitosis takes place, and you know that at the end of the mitosis, still they are same diploid cell. After the growth also, they are same diploid cell. But meiosis, even the completion of the first meiotic division, they become the haploid cell. And the completion of the second meiotic division, they become the truly haploid cell. They are also N, but they are truly haploid. The content of the DNA is also equal to the content of a haploid cell, wherein after the first one, the content of the DNA is little more. So it is equivalent to a diploid cell, but then after the separation of the chromatids, then it will really become truly haploid after the end of the second meiotic division. Now what we call, as long as they are multiplying, we call them the mother cell. So whether we can call them as spermatogonia, gonia means mother cell, or we can call them as oogonia, sperm mother cell, or egg mother cell. After the growth phase only, we call them as primary gametocyte. So primary gametocyte, whether in case of the males or female, the primary gametocyte, it has not undergone any maturation division, so it is still, okay, it is still 2N. Now, after the completion of the first meiotic division, after this, so first meiotic division, after that you will have the secondary gametocyte. Now gametocyte means, I am just writing in general, gametogenesis. So if it is spermatogenesis, we can call it a secondary spermatocyte, and if it is oogenesis, we can call it a secondary oocyte, like that. And secondary oocyte or secondary spermatocyte is the first haploid cell, because it has born out of the meiotic division in the primary gametocyte. The primary gametocyte was a diploid cell. Then after that, after the completion of the second meiotic division, in case of the males, we call it as spermatid, and in case of the female, we can call it as ooted. It becomes continuous in case of spermatogenesis, but it is not continuous, it is discontinuous in case of oogenesis. Are you getting all these points which I am saying, that continuous and discontinuous? Nothing is goofed up. Sir, sloga chapati. What is that? Shall I speak slow or faster? What do you think? Click the cursor. Thank you, beta. I was needing these type of chinna helps only. What happens now? Normally, marathon happens once in a year. So there is a psychological pressure of that. So the one who takes classes every day, 6 classes or 7 classes on an average throughout the year, still you find that it will. So now the quote is off. AC is not working as it is supposed to work. According to this is power chill. Power chill is this person is sweating. Very good. No worries. We are still blessed. Okay, beta. So this is the general point. So what I was saying, the difference is in case of spermatogenesis and oogenesis, the difference number one is this only, that this is discontinuous. Discontinuous means that it is interrupted 2 times. Once it is interrupted in which stage, and second time it is interrupted in which stage. Now, this we have to answer, and now you have to recall this. And listen to the question carefully, because many a times we know, but we don't understand the question carefully, so that's why. Okay, meanwhile, I will call him so that he can. This is. Okay, marathon. Human reproduction. This was going on. Okay. Now we can do so many things. We can easily zoom it here. If this can open in your classrooms, I think it will be better. Let me just ask him. He has not returned back. Okay. Now we have this board also. Discontinuous, arrested 2 times, once in the fetal life, and once during menstrual cycle. The first arrest is still their primary spermatocyte, okay, because the first meiotic division is not over. The first meiotic division was arrested in the prophase first stage. So in which stage? It is prophase first, arrest. Arrest means stopped, interrupted. In the prophase first, if someone asks you which substage, you can say, sir, it is diplotin. In the diplotin stage of the prophase first, it is interrupted for the first time. And so when the meiosis 1 is not over, you remember what it is called. It is still the primary gametocyte. So in case of female, it is primary oocyte. So they are still primary oocyte. You know that they remain in primary oocyte in that arrested state only, and the birth takes place. And after the birth, through the initial development, initial days of growth, so no change happens, the status quo maintained. But at puberty, at menarche, the beginning of menstrual cycle, and in that menstrual cycle, this diplotin arrest is over, and now it go further, right? So diplotin tarvata diakinesis, tarvata that metaphase first, and then tarvata anaphase first and telophase first. right? So during the menstrual cycle, it completes the first meiotic division. Recall, during the menstrual cycle, it completes the first meiotic division and it becomes the secondary oocyte. Secondary oocyte. Now, secondary oocyte is a haploid cell, but then again in the meiosis second, because meiosis second was going on, but in the meiosis second again arrest happens at the metaphase 2. At the metaphase second, arrest happens. Okay, and in this stage only it is ovulated. That means it come out of the ovary in this stage only, and then it is taken up in the fallopian tube and everything status quo. Status quo is a word used for as it is, no change, like it is as in the same stage, that is metaphase second. And what do you call it? You can call it as secondary oocyte. It is secondary oocyte, right? We don't call it as ooted because the second meiotic division is not completed. It is still arrested in metaphase second. If thankfully one sperm enters into this ovum, then it will initiate the completion, it will overcome the arrest, and again it will cause the telophase second, and at that time it is rendered as the ooted, and the nuclear content comes out as the second polar body. Please remember that in this case the meiosis is also different, and the meiosis is unequal type. Unequal type of meiosis means that after the completion of the meiotic division, whether meiosis 1 or meiosis 2, the two cells are not equal, right? So the first difference is, it is in case of spermatogenesis, it is continuous. It begins at puberty. It begins at puberty, right? And usually completes, it goes throughout life. But in this case, it is little different. The second difference is it is equal type. The meiosis is equal type. And in this case, it is unequal type. So what unequal type means? The unequal type means that one of the product is the cell and one is the polar body which contains the the single nuclear content. The single nuclear content comes out as polar body, right? This is called, it is unequal type. After the end of the first meiotic division, it becomes as secondary oocyte, and the first polar body PB1. PB1 means polar body first. And after the completion of the second one, it is then rendered. Rendered means it becomes ooted plus the PB polar body number 2. So this is the unequal type which we are referring to. Any other difference in oogenesis and spermatogenesis? Yes, I can tell you. I cannot wait for you to answer. I can tell you, right? And the difference here is, in the case of spermatogenesis, there a lot of differentiation happens after the maturation division. I mean to say that the structure of a spermatozoa and the spermatid is entirely different. Entirely different. So it undergo differentiation, and that differentiation phase we call it as, what do we call it? Spermiogenesis. And in this case, it won't undergo any such differentiation. It does differentiate. We cannot say that it is zero, but the type of differentiation that happens in the male gametes, that is not happening in the female gamete, right? I mean to say, the ooted structure and the complete, when it is ready to fertilize, the nucleus is ready to fuse, it is still the ooted only. It does not undergo any differentiation. But here spermatid undergo differentiation and it becomes the spermatozoa. Am I clear to everyone? Now I can just take some water. Okay. I am also thinking. Oh my gosh. Hot water. Hot water. I'm already hot. Yes, many people are recalling the trick also. Very good. हाँ बेटा. Call the operator. Let me make a call. We know students are calling. Students are calling. Some issues are there. Can you come over here? Please come. I called him. I think yes. He's busy in some. I think eating some breakfast. Right. So I think, so one more thing I want to share here, that these are the stages of the egg. Stages of the egg are oogonia. Okay. So oogonia, then it undergo multiplication by mitosis. Still it is oogonia, that is the germ cell. After that, after the growth phase, it becomes the primary oocyte. Primary oocyte. Primary oocyte is still the 2N, it is no change in the ploidy level. Then it undergo the meiotic division. The first meiotic division, it becomes the secondary oocyte. Secondary oocyte, and this is the first time it becomes haploid, and then it becomes the ootid after the completion of the second division. Okay, and it is truly haploid ooted. So these are the various stages here. Now please understand that at the time of birth, a female is born with many million, a couple of million, NCERT book writes, a couple of million primary follicle. Now, primary follicle, the word they use is a follicle, the group of cell. कुछ बच्चा लोग कुछ यूज कह रहा क्लास के ड्यूरिंग वो कॉल कर रहा है मेरे को. कोट ले जाओ बाबू, कोट ले जाओ, इसी काम नहीं करना. In any class, some issues are there. हाँ बेटा, we can fasten from this because in the beginning, little tuk and then we play the shots. हाँ बेटा, we can fasten from this because all classes, no issues, no issues. Okay, I hope bachcho, this is, so now the follicles, a female is born with primary follicles. So primary follicle is a group of cells surrounding the primary oocyte. So primary oocyte surrounded by granulosa cell. The primary follicle will then grow into a secondary follicle. In a secondary follicle, it is still in the primary oocyte. This is primary oocyte. Primary oocyte. Now what happens? The change happens in secondary follicle. Okay, the secondary follicle, it still is primary oocyte, but many layers of granulosa cell. And then a theca means wall. A wall start developing outside. And theca start developing, not fully developed, start developing, and many layers of granulosa cell. And this is still it is primary oocyte. It is undergoing the further, diakinesis and then metaphase first. All these are going on in this time, but it is yet not completed. After that, then it becomes the tertiary follicle. Now, tertiary follicle is a follicle with lot of changes, and the biggest change in tertiary follicle is the cavity, colantrum. And it is at this stage, NCERT textbook write, it is at this stage, the primary oocyte will become the secondary oocyte. The secondary oocyte, and then the first polar body, so polar body number 1, PB1. And this is antrum, and one more change the textbook write, and that is the theca is differentiated into inner cellular theca interna and outer cellular theca externa. Okay, it is called tertiary follicle. Tertiary follicle characterized by antrum. If only one change, you have to answer. And tertiary follicle will grow further, and now it is a completely developed Graafian follicle. Graafian follicle, and no any change. The biggest change is size. It becomes bigger only, and the change which you have to remember, which is completed by this time, is a non-cellular layer is formed all around the egg. And you know the name of this layer. The name of this layer is called ZP, and ZP stands for zona pellucida. Zona pellucida. Rest of the changes are like same. These are the granulosa cell. This is antrum, theca interna, theca externa, and this is still secondary oocyte. So these are the stages of the follicle. So बच्चों please remember follicle के stage क्या हो गए? Primary, secondary, tertiary and Graafian. Primary, secondary, tertiary, Graafian. Remember 4. Now in the first two, primary and secondary, it is still primary oocyte, and in the second two, that means the tertiary follicle and the Graafian follicle, you will get inside is the secondary oocyte. The first polar body is also released at this time only, right? And please understand that in this time it is still inside the ovary. Now here, one more thing you have to remember, it is producing a hormone also. As the theca develops, theca produce estrogen. Theca become bigger, okay? More estrogen. Theca become further bigger. More estrogen. Such a high level of estrogen that this estrogen is enough for the change in the GnRH pulse frequency, and this results into an LH surge. This results into an LH surge. And LH surge, now the LH means luteinizing hormone, and the purpose of this hormone is to make this as corpus luteum. It cannot become corpus luteum without this removed out of this Graafian follicle. So the first thing that it does is rupture of Graafian follicle, rupture of the outer layer of the ovary, and oocyte is released out of the ovary. This is called ovulation. And then where is the egg right now? At this moment, where is the egg? It is lying in the coelomic cavity. And from here, the fimbriae takes it inside, and it goes inside the fallopian tube. And the rest of the cell will turn into a yellow body. The yellow body, it turns into yellow body. This yellow body is called corpus luteum. Corpus luteum. Corpus means body and luteum means yellow. Corpus luteum. And this is happening because of LH. And corpus luteum is also growing. That's a tendency. And remember that corpus luteum is a endocrine structure. It is producing progesterone, and as the corpus is growing further, it is making more progesterone. Plus, it is also making some estrogen. So you find that there are two peaks happens in the luteal phase. One peak is of the progesterone, other peak is of the estrogen. So also estrogen you can find out, you can find coming out of this corpus luteum. Therefore, NCERT writes that few hormones, ovary produce few sex hormones, right? The testes produce one sex hormone, but here few sex hormones. I hope you understand that. So progesterone and estrogen, these are the two main hormones which are produced in the luteal phase, and so you'll find the two peak happening. Initially the peak of the progesterone and then another peak of estrogen. So estrogen will undergo two peaks, that means highest level, two peak of estrogen during the menstrual cycle. I hope you understand all this, right? Now what happens that such a high level of progesterone, the hypothalamus does not like that, and hypothalamus will stop the GnRH, and because of stopping the GnRH, the LH is also withdrawn. Now what will happen? The pituitary will not release LH now, and LH level in the blood is decreasing. And as the LH level is decreasing, the corpus luteum is also shrinking. And the corpus luteum is shrinking, the level of progesterone and estrogen are also both coming down. And this corpus luteum keeps on shrinking, and it is completely degenerated, and it becomes a white scar called corpus albicans. Okay, corpus albicans. Okay, so what has caused the change in the GnRH pulse frequency? The level of progesterone, the level of estrogen, the level of testosterone, the level of relaxin. So the answer here is the level of progesterone. But I have seen one question which says that the level of estrogen, the level of progesterone, and third option says the level of both estrogen and progesterone, and fourth one is something else, right? Now here in this case, you have to write that both high level of P plus E, both high level of P plus E are causing the change in the GnRH pulse frequency. Do you remember which gland is producing this GnRH? Okay, so change in the GnRH pulse frequency, you will find the LH is withdrawn. This is called the negative feedback mechanism. LH is withdrawn, and because of the withdrawal of the LH, corpus luteum shrink, and because of the shrinking of corpus luteum, the P plus E both coming down. Now the P level, that is progesterone level, comes very, very low, and at such a low P level, what will happen that the endometrium cannot sustain. Remember that endometrium to remain there and also remain happy and secreting, it requires progesterone. So if progesterone is not there, endometrium will undergo ischemia. Ischemia means the stoppage of blood supply to the endometrium that causes the shedding of the endometrium, and some capillaries open up at that time, which causes some bleeding, right? And so this is known as menstruation. The menstruation, the bleeding phase of the menstrual cycle is called menstruation, and this lasts for somewhere 3 to 5 days, 3 days to 5 days. Okay, bachcho, you understand this point, what I'm saying? So I have linked the oogenesis with the menstrual cycle, right? And in case if any issue is there, you can just write. Meanwhile, I will wipe off my... Haan, beta. Very good. Progesterone. Only one if you have to answer, then progesterone. And if you have to answer, like if there is an option called P plus E, then obviously P plus E both will give the negative feedback. And you will find that because of the negative feedback, as the corpus luteum shrinks, the both level P plus E comes down. Okay, when we open those NCERT pictures, at that place we will see this. Okay, bachcho? So what I am saying here is, you can notice here in the menstrual cycle diagram, this diagram. So the hormone which undergo two peaks, one is called the estrogen undergo two peak. Answer is estrogen. First peak is in the follicular phase, and the second peak happens in the luteal phase. Remember that progesterone is only having one peak, and FSH and LH also having single peak. The peak level of LH is known as the LH surge. So इसमें दोनों में कौन सा ऊपर वाला पाइने higher one, this is called LH surge. LH surge. Please remember that this happens in the middle, roughly in the middle, roughly little before that, little before, one day before, to be too true, I can say one day before, and you will find this at the same time the peak of FSH also seen. So the lower one, the red one, is of the FSH, the blue one is of the LH. How to identify? Paine is LH and Kinda is FSH. And here two colors are there, blue and red or blue and pink. How to identify which one is estrogen and progesterone? If there are two peaks happening, then it is estrogen, and if there is only single peak, so clear graph, single peak and then down, this is the progesterone. And you will find that as the LH is withdrawn, when the LH is withdrawn, you will find LH jab withdraw ho gaya, you will find the shrinking of the corpus luteum. Now this is the time when the corpus luteum is really very big and it is producing lot of progesterone plus estrogen, and very high level also. The hypothalamus does not lie and stop the GnRH, and because of the stoppage of the GnRH, the LH is withdrawn. Yes, I answered my question myself only. I asked you which gland is producing GnRH, and just now I spoke it that it is hypothalamus. So hypothalamus produces gonadotropin releasing hormone. In response to that, the LH, is secreted. So if the GnRH pulse frequency changes or comes down, then LH is also withdrawn, and this is called the negative feedback. How to identify it as a negative or positive? You can see that when the level of progesterone is very high, so the feedback is to make it low, right? So now because of this feedback, the level of progesterone comes down. You understand? Level of estrogen comes down. One is growing, and this is causing this to fall, right? So this is happening in opposite direction. So high level of progesterone will ultimately become the low level, and this happens because of the low level of the GnRH. So if the cause and effect are in opposite direction, it is known as the negative feedback mechanism. Negative feedback mechanism, remember that all homeostasis in our body is due to negative feedback mechanism. Why I am sweating right now? Because inside the body has more heat, more calorigenesis, and therefore it is giving a negative feedback to my hypothalamus, and as a result of that, there is more. Hypothalamus is asking the sweat glands, sending signals to the sweat glands, ki bro, please produce more sweat, so that the sweat, because of the sweat, because of the evaporation of the sweat, we can cool this body, right? So this is also an example of negative feedback. Why it is called negative? Because high level of body temperature is ultimately causing its getting lowered, and therefore it is in opposite direction, and therefore it is called negative feedback. So temperature regulation is negative feedback. Any regulation in the body, any hormone level regulation is also an example of negative feedback. Osmoregulation is an example of negative feedback. Remember, all homeostasis is due to negative feedback. Bachon, the menstrual phase, the 3 to 5 days menstrual phase, then comes the follicular phase, and tarvata is the luteal phase. Now in this time, the luteum is really very big, and because of that, the endometrium glands are really very much developed, and lot of fluid is being secreted, which is ultimately causing or helping the blastocyst to implant, right? But as you see that if the level of progesterone comes down and estrogen comes down, the same change also seen. So the growth of this is also going down. Why? Because the cells are shedding, and this shedding of the cell is known as menstruation. Please understand that the luteal phase is usually of 2 weeks. 2 weeks and 14 days. 14 days, usually of 14 days, the luteal phase, right? So in case if the menstrual cycle is not of the 28 days, it is of, let us say, 31 days. 31 days, right? But then still the luteal phase will be only 2 weeks. So there is increase in either in the menstrual phase or in the follicular phase. Or let us say if the menstrual cycle is not of the 28, 29, it is of 20, let us say 27 days, right? But in a 27-day menstrual cycle also, the luteal phase will be 2 weeks, 14 days, and there is a decrease in the days of either the menstrual phase or in the follicular phase. Got the point? So can I say this line is a line, if this line comes as an assertion that the ovulation happens 14 days prior to menstruation. Ovulation happens 14 days prior to menstruation. Now, how to understand this line, what the teacher is saying or what the question is saying? The line is saying that ovulation 14 days prior. Prior means mundu, before, prior. Okay, prior, pre hota hai na, pre, before. So 14 days before the ovulation occurs, 14 days before menstruation. So यहाँ पे menstruation हुआ and यहाँ पे ovulation हुआ, right? So it is happening 14 days before the menstruation. So the assertion is correct that ovulation happens 14 days earlier to the menstruation, right? And then the reason is given as the luteal phase lasts for roughly 2 weeks. The luteal phase lasts for 2 weeks, and this makes the answer as both assertion and reason are correct, and reason is an explanation of the assertion. Make sense? Got the question? Right? So these questions only you will get one test based on what we are discussing right now. This is a menstrual cycle that just now we have seen, and you will find that because of the shrinking of the corpus luteum, the level of progesterone is coming down. But what happens if some implantation happens? Now, implantation happens, that means the blastocyst is inside. So blastocyst is here, and blastocyst will not let this shed, and so that because if this sheds, the blastocyst will be out of business, wo bhi chale jayega, right? So therefore they will cause outer layer will actually produce a hormone which is known as HCG. Outer layer is called chorionic layer, and therefore the name of the hormone is human chorionic gonadotropin, and human chorionic gonadotropin is analogous to LH. Analogous to LH means it is same as LH. It is also working like LH. It is binding to the LH receptors, and if it binds to the LH receptors, right, the corpus luteum. So here you have the LH receptors, HCG binds to it, and HCG again revives it. So rather than becoming corpus albicans, it again grows into corpus luteum. It again grows into fully developed corpus luteum because of this. I hope you understand this. Because of the HCG, it is gonadotropin, therefore it is an LH analog. LH is a gonadotropin. Okay, bachcho? And this is the reason that why during the pregnancy there is no menstruation. Why there is no menstruation? Because, right? Why there is no menstruation? Because there is no fall in the level of LH and FSH. Why LH falls, but LH, duplicate LH called HCG, that level is there. And who is producing the HCG? The implant, the implantus or the early embryo is producing that. This is showing the fertilization. Okay. Then remember that blastocyst is a two-layered structure. Outer layer is called trophoblast, and inside all the cells are collected at one place rather than making a full layer. All the cells are collected to one place that is known as inner cell mass. Remember that all the cells of the inner cell mass are totipotent, or they are the stem cell. They are the one who can produce the entire body. Yes, entire body. It means that if I am having right now 10 inner cell mass, 10 cells of the inner cell mass, and suppose if I have a technique to grow all these 10, so all the 10 are going to make the same body. All the 10, because all these 10 have the information to make the entire body. And if I introduce any gene in these cells, right, in these 10 cells, if I introduce the gene, remember that they are going to make the entire body. And therefore, all the entire body will be having, all cells of the body will be having that gene. And therefore, they are trying that if they can anticipate some genetic disease before birth. So in this time only, in the embryonic stage, if the gene therapy is done, gene therapy matlab gene replacement or gene, we can give a gene. In that case, what will happen? The whole of the body will be having that gene, and we don't require, again and again, you have to infuse the lymphocyte, as happens in the case of the SCID, severe combined immunodeficiency, ADA deficiency. Okay, bachcho, now please understand here the different stages of the embryonic development are given. Let me just recall. First month, at the end of the first month, you will have the heart. At the end of the first month, you will have the, suppose if this is all 9 months, so first month somewhere here, okay? So you will have the heart beating, heart start pumping, heart start beating. So the first functional organ is the heart, but first differentiating organ or structure is the CNS, the brain and the spinal cord. They are the first differentiating structure, but the first functional structure is your heart, no doubt about that. And what happens at the end of the first trimester, what happens at the end of the 5th month, and what happens at the end of the 2nd trimester. In the 3rd trimester, thankfully, we are not, we don't have to remember anything. All this will be seen here. Okay, so let me ask you, first month, heart, 2nd month, limbs and genitalia. Now 3rd month, that is first trimester, limbs and fingers, digits are fully developed, and this is the time when the genitals are also developed. The beginning of the growth of genital happens here, and at this time the genitals are also grown, and we can of course see the genital by ultrasonography. And therefore, if they can find out any male structure or the penis, then they can say that this is the male fetus. You understand my point, right? So this is this time by sonography. At this time, you can say it is male or female. However, it is a crime in our country. It is a crime to to do the prenatal sex determination. But in other countries, it is mandatory. Okay, bachcho. So I hope you understand here. So most of the organ systems have started developing, and limbs and digits are completely developed. Initially, it is just like these digits, but here now the digits are also developed, and genitals can be seen growing at this time. The 5th month, you will find that the first movement happens in the 5th month. 5th month, okay? And first movement happens, and also the hair on the head. Now at the end of the second trimester, okay, so let me write here like T2, trimester 2 complete. So here you will find that most of the organ systems have been formed by this time, and also you will find that the fine hair all over the body, fine hair, this is hair on the head, but here fine hair all over the body, which remains, even after birth also, in most of us at the time of birth, they were there. And in the first few months, when our mothers, parents are giving us the bath or giving us the massage, at that time, these hair will shed. So yes, these hair, they develop by the end of the second trimester. The end of the 3rd trimester, the complete development is this. The whole duration is 9 months. 9 months. Okay? Okay. We're coming to this almost end of this chapter. Roughly 4. The first R is not very productive. In terms of, we only completed 1 chapter. But, okay, anyways, because of course, the batsman also comes, even good batsmen, they come, and the first few overs, they just play little slowly, and then they will increase the speed. You also, your metabolism also developing, and initially, you also takes time so to maintain that. And now, 5th month, first movement, very good. 5th month, first movements, okay, very good. Now, bachcho, let's come to the reproductive health. Now, this chapter is, we are expecting 3 questions out of this. And these 3 questions, okay, if we are lucky enough, we can get the questions like, when is the family planning program initiated, in which year, okay? So just remember that 4 years after the independence, you will find this family planning program initiated by the government of India. Later on, this becomes the RCH program, later on the same family planning program. So family planning program 4 years after independence, like that I remember, you can also remember the same. So it's come out to be 1951. Later on become the RCH, Reproductive and Child Health Program, with more of these areas to take care of, not only family planning. Family planning matlab maintaining a small family, hum do hamare do, right? Hum do hamare do. Okay, now the population stabilization and birth control, all these data are just the old data, so no need to take any tension about that. We don't have the latest data. Latest data not available in the NCERT, but I can tell you the latest data. The population of India as per the 2011 census, as per the 2021 census, is 140 crore or 1.4 billion. 1.4 billion. The population of India as per the 2021 census is 1.4. As per the 2011, 1.2, but 1.4. Okay, bachcho. Now, this is there. Achha, one more thing. The growth rate is not increasing. Population, no doubt, increasing, but the growth rate is decreasing. The growth rate is decreasing. It was in the 1980s, it was more than 2. And it is well below 2 now. Although it is marginal, but still we cannot say, like if the question comes that both population growth rate and the population grows in India in the last, let us say, last 50 years, both population and the population growth rate increase. Wrong. Population increase, but the population growth rate, annual average growth rate has decreased. Please understand that. Reason for increasing population are these, easy. Steps to overcome, also easy. Okay, we are lucky enough we can get all these. Now, contraceptives. Contraceptives, now we have the natural method or traditional method. Now here a question came. Honestly, I'm telling you, this is not my question. Honestly. If a teacher is saying honestly, that means he is very honest. Otherwise, he would not have told. Right. Otherwise, I would have kept it in my heart only. The question came yesterday is about the traditional method. It says that one of the traditional method called coitus interruptus, and this is a contraceptive method, contraceptive. And the reason gives that the male partner withdraw the penis and therefore it is not letting the semen to enter into the vagina. Yes, this is the reason. I mean, why it is called contraceptive? It is called contraceptive because it does not allow the fertilization and the implantation, that is pregnancy. It is contra inception or contraception. Inception ka ulta, contraception. I hope you understand that. So it is also known as traditional method, natural or traditional method of birth control, periodic abstinence, also known as the calendar method. Please, bachcho, remember, I think you are just carried away by the name traditional. So please remember, another name of periodic abstinence is calendar method, and another name of calendar method is the rhythm method. Rhythm method, wherein some fertile days and some unfertile days. Unfertile period is a safe period. Safe period means no pregnancy will happen. So that is called the safe period. So now you have the safe period and the fertile period. So fertile period, yaad kar lo, fertile period is the first 7 or 18 to 28. Okay. No, 1 to 10. Because 10 to 1 to 9. 1 to 9 is a fertile period, and 18 to 28. This is also the fertile period. So the safe period will be from 10th to the 17th day of the menstrual cycle. It's called the safe period. 1 to 9 and 18 to 28, these are the fertile period. Okay, bacho? So please don't get carried away by just these terms. I think right now I have seen in many students, most of the students, right, that they are not making mistake. If I change the question language when I am talking to them, because at that time only one question is focused, that is also a difference. At that time the paper was over because we are meeting after that paper. So another factor is this: there is no constraint of time here, there is no pressure of time. So yes, one thing is this also. And the moment I make a small change, and then you can give the answer. That means it is not the concept or the memory loss, you forgot anything. It is like you could not understand that language, and or the question. Language does not mean that only difficult words are there. Language means how the teacher has placed that question. That is also the language of the question. Okay, bachcho? For example, like yesterday, some people have said, sir, iska answer to hona chahiye ki it is not a explanation about the traditional. And very good students. I said, okay, beta, Nikhil, just listen. I am saying to you, I am now doing one change. Okay, now listen. I just changed the word traditional to natural. That one of the natural method of contraception is coitus interruptus. And then I say, I just read the reason. And then I asked him, what's the answer? Sir, answer is now 1. Okay, because at that time the word traditional was not understood. They are thinking that traditional matlab traditional, old, right? It is of no working today. So it is not traditional or advanced in any, it is just another name of the natural method. Then We have other artificial methods which include the barrier method, the IUDs, the oral contraceptives, the injectable, the implants, and the surgical method. Let us go from down to up. Surgical method, very easy, tubectomy and vasectomy. Okay, implants, injectables, and oral contraceptives. That means hormonal pills. All three have the same mechanism. Only the way the drug is given to the body or the hormone is given to the body is different. In oral contraceptive pills, tablet is taken. Injectable, depot injections. Depot injection means where the injection is given in the fatty tissue in such a way that all the drug is not released at that time. It is there in the fatty tissue, and little by little, daily, some amount of drug is now coming into the blood. So this is called injectable. Generally, we can give injection for up to 6 weeks. Okay, 6 weeks. So up to 6 weeks after one injection, there is no need for any tablet to be taken. Then comes another advanced method is implant. Now we have the latest implant, the Norplant. You have seen the diagram of that, the 6 needle-like. Actually they are silicone, so they are very, very soft also. The 6 needle-like, which is impregnated with the hormone, the same drug, and that can give the contraception for up to 5 years. So mechanism of action is the same, but only thing happened is duration has increased. I hope you understand. So oral contraceptive pills, which is hormonal, injectable, and implants, all three same hormone, that is estrogen and progesterone and estrogen. I have to speak the progesterone first because the action is of progesterone. But because when you give progesterone, you will not have the menstrual cycles. And if there is no menstrual cycle, there is no follicular development. And if there is no follicular development, there is no estrogen as well. So the body will not show signs of lack of estrogen. That is the reason estrogen is also added. But the main hormone in these all three methods is the progesterone. Okay, but now you will wonder that, sir, progesterone, it is a pregnancy hormone now? Yes, it is pregnancy hormone. But hormones, when it is taken and how much it is taken, that can change its entire effect. So when and how much, that means a dosage, that can change. In fact, the GnRH, the same hormone GnRH, only the pulse frequency differ. If in a different pulse frequency, pulse matlab the peak, in every minute higher level happens, or every 10 minutes higher level happens. So every 10 minutes frequency is lesser, every minute frequency is higher in these two comparisons, right? Like that. So only the GnRH pulse frequency that can alter its effect. So either FSH to increase in the blood or LH. It depends on different GnRH pulse frequency. So, bache, in endocrinology, this is very, very important that many of these hormones have got overlapping effects, number one. Number two, hormone effect can change if the dosage and the course is also changed. Okay. So now what are the different, mechanisms? Please remember 3 mechanisms in these hormonal method. That means pills, injectables, and implants, PII. So what are the 3 things that the hormones are doing? Number one, the hormones are making they will stop the ovulation, withdrawal of ovulation, number one. Number two, they are making the endometrium unsuitable for implantation. And number 3, they are changing the thickness of the cervical mucus, which will turn into a mucus plug. And mucus plug means that the passage for the sperm is stopped. It is no entry for the sperms, right? 3 things happening. Number 1, what was that? Stopping ovulation. Number 2, what they are doing? Making the endometrium unsuitable for implantation. And number 3, it is making the thickness, the consistency, the thickness of the mucus changes. As a result, the mucus becomes thick. It becomes a thick mucus plug. Okay, bachcho, got the point? 3 things. Now, IUDs are of various categories. The first generation are non-medicated one. That is, an example is Lipiz loop. It is just made up of plastic. Then second generation were the copper, copper releasing IUDs. Copper releasing IUDs, they are copper T and copper 7. One more copper releasing, do you remember the name of one more copper releasing IUD? One is copper T, copper 7. They are very easy to understand or identify. But please understand, yes, I have seen people are answering. Why you, deleted the answer? You were right. Yes, it is multilode 375. Very good. Multilode 375 is another copper releasing IUD. Very good. And so copper releasing IUD, the copper ion, copper ions in the uterus, they are making the movement of these pumps. So it is retarding the movement of these pumps. The movement of the tail of the sperm is retarded. As a result of that, the fertilizing ability of the sperm is also being taken away. The sperms become incapable of fertilization because their speed, their motility is changed or decreased. Very good. That is a copper ion effect. What was the Lippi's loop only doing? See, if any foreign body is inside the uterus, whether it is Lippi's loop, copper T, or any hormone releasing IUD, any such thing, any mechanical device inside the uterus, it is increasing the phagocytic activity of the of the phagocytes, the macrophages. So phagocytic activity increases. There is a local inflammation continuously happening. And because of the local inflammation, two things are happening. Number one, the phagocytic activity, the sperms are being phagocytosed. And number two, because of the local inflammation, there are many chances that the implantation cannot happen in an inflamed endometrium. And why local inflammation happening? Because of this foreign body inside the uterus. So whether it is Lippi's loop or it is copper releasing or hormone releasing, two of the effects are common to all three of them. That is, number one is local inflammation, phagocytic activity, and number two, that taking away or decreasing the ability of the implantation. Now coming to the hormone releasing IUD, do you remember the name of the hormone releasing IUD? First of all, remember which hormone they are releasing. Again, the same hormone, progesterone plus estrogen, the same tablet wala hormone. The tablet wala hormone is inside the body, that means absorbed in the blood, and now this hormone is in the uterus. Same hormone, progesterone and estrogen. Very good. So the two hormones are same, and can you recall the name of the hormone releasing IUD? And that is LNG20. Very good. LNG20 and progestasert. Very good. But right now I'm not giving the shout out. You can understand that in the beginning the speed was low. The target was not fulfilled. Therefore, let me quickly cover it. Okay, bachon, so the two things the hormone releasing are doing in addition, and the two things are, number one, they are making the endometrium unsuitable for implantation, and number two, they are making the consistency of the mucus change, and therefore it become a thick mucus plug, and which will not allow the sperms to enter into the cervix. Okay, so I hope you understand the difference of the effect of hormone, whether it in the IUD, same hormone, but the effect is only two, that is the local effect, endometrium unsuitable and thick mucus plug. But when the same hormones are taken inside in the systemic circulation in the blood, now it is doing another third change, and now it is also taking away the ovulation. Okay, so it will stop the menstrual cycle, and because menstrual cycle stops, therefore there is no ovulation. Okay, bachcho, all well. IUDs. Yeah, the pills. Now pills, number one, we have the hormonal pills which contain progesterone and estrogen combination, or simply POP. POP matlab progesterone only pill. And number two is the Saheli. It is also a tablet, it is also a pill, but it is not hormonal. So which hormone it is not there? The two hormone, progesterone and estrogen, is not there. And these are the steroid hormone. You know that these are steroids. So we can say that the Saheli is a non-steroidal tablet, whereas these are the steroidal. Why steroidal? Because these hormones are actually steroids. Very good. So non-steroidal or non-hormonal tablet, it contains a chemical called centchroman, and this is only taking away the implantation ability. And how it is changing the implantation activity or taking away the implantation activity by making the uterus, the endometrium, not responding to the estrogen. So the estrogen receptors are blocked. The local estrogen receptors are blocked inside the endometrium, and because of that, it is causing this. It prevents implantation. It has few side effects and it is high contraceptive value. And more than that, it is developed in our country only. It is Swadeshi. Swadeshi, that means it is our own country. It is CDRI, Central Drug Research Institute, Lucknow, developed by them. Okay. Coming to the MTP Act 1971, MTP Act 1951, which act? Not the act, but the family planning program. But 1971, the act, this is called the MTP Act, Medico Legal Termination of Pregnancy Act. Now, every act is being, they are time to time, they are updated. And one of the latest, and whenever the acts are updated, we call them amendments. So one of the latest amendment, which is very, very important, which has really a big change happens, and that is in the year 2017. In the year 2017, the big amendments happened, and in that case, what happens now? In place of one doctor, two doctors' opinion are required. Only thing that changes, initially only one doctor was enough to certify that yes, I am saying it really required, it is a real candidate of MTP, but now the opinion of two doctors required, so chances of misuse are less. So what are the probable misuse of MTP? See, the MTP is making the people very casual about contraception and about the pregnancy, and therefore people are doing these MTPs without any certificates or without any legal control, and therefore a lot of people who are really not trained doctors who are doing this MTP, and in any stage they can do, in any trimester, their main target is the money, but then in few days we find that the person become really very ill, and then she dies. So therefore this is being misused. But thanks to this MTP, the termination of pregnancy is now legalized, and now thanks to this amendment, which is now two doctors required. So two doctors, chances of false certificates are little lesser. I'm not saying they are not there, but now two permission of two doctors, opinion of two doctors required. So MTP is performed to get rid of unwanted pregnancy. It is induced abortion. MTP is just legalized induced abortion. And it can be done in the first trimester by any of these, either the failure of contraceptive, or if continuation of pregnancy could be harmful or even fatal to the mother or the fetus, or it is either due to casual unprotected intercourse, or there is a substantial risk that if the child were born, it would suffer from severe physical or mental abnormalities. So the first trimester, it can be done easily. It can be done easily. But then comes the second trimester. The second trimester, that is from 12 weeks to 24 weeks. In that trimester, two doctor opinion required. So second trimester, two doctor opinion required. And also the opinion is that really either the mother is at risk or the child born will be having probably a lot of physical and mental abnormalities. So that chances, because it is becoming riskier as compared to the first trimester. In the second trimester, it is riskier. First trimester means the first 12 weeks. And then it becomes in the next 12 weeks, that is T2, it is little riskier. And T3, it is really very dangerous and it should not be done. Okay, so in any case, in any of the possibility, it cannot be done. And if it is done, it is a legal, it is a criminal offense. It is an act, okay, Qanun, act. Then STDs, there are several STDs, gonorrhea, genital herpes. Now identify the bacterial one. The syphilis, the gonorrhea, and chlamydiasis or chlamydiasis. Okay, these are the bacterial one. Syphilis, gonorrhea, and chlamydiasis. The viral one are genital herpes, genital warts, AIDS, and hepatitis B. So 4 names are there for the viral one. Another name is another viral one is here, the genital warts. So total 4: genital herpes, genital warts, and third one is AIDS, and fourth one is hepatitis B. Right? So we know that this name of the hormone is easy. Only problem happens in case of these two: genital herpes and genital warts. Herpes is herpes simplex virus. So it is very simple because herpes name is coming. So now only one name to be remembered, and that is genital warts. Genital warts is HPV, human papilloma virus. Human papilloma virus. Genital warts, human papilloma virus. Genital warts, human papilloma virus. Okay, clear? Now please remember that AIDS, hepatitis B, and genital herpes, these 3, AIDS, hepatitis B, and genital herpes, these are not completely curable. Okay, AIDS, hepatitis B, and genital herpes are not completely curable. Now, do you remember any other organism which is causing the MTP, the STI? Another organism is a protozoan, and what's the name of the protozoan? It is Trichomonas vaginalis, the name of the disease, trichomoniasis. Okay. So what are the complications of the STIs or STDs? The complications of the STIs are I space, okay? Okay, I space, that means I stand for infertility, S stand for stillbirth, P stand for pelvic inflammatory disease or PID, A stand for abortion, C stand for cancer of the reproductive tract, and E stand for ectopic pregnancy. So this is what is I space, okay? I space. So total names are 6. 1, 2, 3, 4, 5, 6. Total names are 6. I for infertility, S for stillbirth, P for PID, A for abortion, C for cancer of the reproductive tract, and E for ectopic pregnancy. So they are also asking the secondary, or they will give the name as complications of STIs. Now, if there is a lack of pregnancy, if the people are not getting they are not fertile, there is infertility. Infertility means they are unable to produce a viable offspring despite 2 years of sexual cohabitation. Despite 2 years of sexual cohabitation, still they are not producing children, then we can consider them as infertile, okay? So infertility also stands for if the pregnancy is continued, but then it cannot be completed, then also it is infertility. So don't think that, sir, once it is implantation happens, it is not infertility. No, we cannot call it sterility, but certainly we can call it as infertility, because the definition is unable to produce a viable offspring. Viable, live birth, unable to produce a viable offspring despite 2 years of sexual cohabitation, unprotected sexual cohabitation. In that case, what we can do now, we can have the test tube baby program. The test tube baby program is actually the in vitro fertilization is called the test tube baby program. In vitro fertilization means the fertilization happening in the laboratory. In controlled conditions in the laboratory, the fertilization is done. Okay, so gametes are collected from the wife or the husband. If the wife is not giving the gamete, then either the donor, or if the husband gametes cannot be used, they're either again the donor, right? So gametes are collected and they are allowed to fertilize in the laboratory in controlled conditions, and then the embryo develops. So zygote becomes the two-stage embryo, two cells embryo, two blastomere embryo, then 2 become 4 and 4 become 8. Up to 8, if you can collect it, then you can transfer it inside the fallopian tube of the wife or of the surrogate mother, right? Or if surrogate mother means the hired, where we are hiring a person only for the surrogacy. That means only for the pregnancy we are hiring. We are actually paying rent for the uterus. This is what is called hired uterus or surrogate mother. Okay, so up to the first 8, 1 to 8, from the zygote up to the 8 blastomeres, we still call it ZIFT. We transfer it inside the fallopian tube, right? It is ZIFT, zygote intrafallopian transfer, and more than 8 blastomeres, it is now simple. It is IUT, intrauterine transfer, intrauterine transfer. Both are known as embryo transfer. Both comes under the ET, embryo transfer. So please remember that all IVF, all in vitro fertilization has to undergo ET. Please understand that embryo development cannot occur in vitro. It has to be in vivo only. It has to be in the live person, right? In vivo, in the living form. It cannot be in vitro. So embryo is always transferred after the IVF. So therefore, IVF full form, in vitro fertilization. What is ET full form? Embryo transfer. What is ZIFT full form? Zygote intrafallopian transfer. And what is IUT full form? Intrauterine transfer. Only if you can remember these abbreviations, I think you can recall everything. With few lines, if we add them, then it is over. Okay, then after the in vivo fertilization, okay, that means if the fertilization happens inside the body only, but this women cannot conceive. Cannot conceive means that the implantation cannot happen, or maybe implantation happen, but then the first trimester or the second trimester, or the abortion happens. So in that case, if there is no implantation, or if the abortions are happening spontaneously, then still we can do embryo transfer. So in that case, we will not repeat the fertilization, because fertilization already happened inside the body. So we will collect the embryo from the body, from the fallopian tube. We'll collect the embryo and then we can transfer it to a surrogate mother, right? And here also same ZIFT, okay, or IUT, same ET, right? If we are able to collect up to 8 blastomeres, and if we are able to transfer up to 8 blastomere stage, then we can call it as ZIFT. In ZIFT, we do it inside the fallopian tube. In IUT, we do it inside the uterus. More than 8 blastomere should not be done in the fallopian tube. It should be done inside the uterus. Please remember that. So yes, ET is also required in cases of fertilization occurring in vivo. That means inside the body. These are the people where they cannot conceive, but otherwise they are absolutely normal. Now, there are some women who cannot give a fertile ovum, right? Or otherwise their hormones are all fine, all the uterus condition, cervix condition, all things fine. But only thing is that there is only the gamete is not coming in the right time and in the right form. Then what we can do, we can collect the gamete from some other person, and this time we can transfer the gamete inside the fallopian tube. We can call it as gamete intrafallopian transfer, or GIFT. Okay. Now please understand that when the gamete is transferred inside the fallopian tube, after that the couple is allowed to have intercourse, and therefore after the insemination the sperms can come and the fertilization can happen inside the ampulla. So where it is happening? Inside the ampulla, inside the body. So we can call it as fertilization in vitro or in vivo. We can call it as fertilization in vivo. That means inside the living body, in vivo. So please understand that in GIFT, the fertilization is happening is in vivo. How? We are only transferring the egg. And when the egg is in the fallopian tube in the ampulla, immediately after that, the couple have the intercourse, and therefore, after insemination, the sperms will come at the place of ampulla and can fertilize. Okay, so that is in vivo fertilization. Now, this is, I think, some people have doubt in this, that sir, how it is in vivo fertilization in case of GIFT? Okay, now in some people, there is some problems are there. Like, problem is low sperm count. The sperm count is low. So in that case, that semen is not good, or it is not giving any fruit. What we can do? We can do the semen wash, and then we can transform the semen, and then we can do the insemination. But when we are collecting the semen out and then processing it, and then again we are transferring it or inseminating, it is called artificial insemination. Artificial insemination, right? Now, insemination can happen either in the uterus or in the vagina, right? If insemination happening in the uterus, it is called IUI, or if it is happening in vagina, we don't have any special name of that. AI, we simply call it AI, or better known as artificial insemination. I remember a joke of AI, that is, the wife is a doctor and husband, no, wife is a engineer, she is a computer engineer, and husband is a doctor. So these days, all the softwares are actually developing these AI, that is artificial intelligence softwares, and AI is really booming up in the next decade, the decade of AI, you know that. Okay, so because she is a professional, they are talking to the people, yes, AI, we can do AI. Her husband was overhearing them. Every time husband hear the wife is talking AI. One day the husband blast off. What is going on behind me? Okay, so well, what you heard? AI. You were talking something about AI. Well, yes, Dr. Sahab, AI. AI here means artificial insemination. For doctors, it is artificial insemination for doctors, and AI for engineers is artificial intelligence. Okay, so AI is same, the meaning may differ. Can you recall? Not the joke. Not the joke, but these abbreviation. IVF, in vitro fertilization. Remember that for in vivo fertilization also, actually the abbreviation is IVF coming only, but then we don't use IVF for in vivo. In vivo, we write in vivo. In vivo. So because in vitro IVF is actually meaning in vitro only. So that is in vitro fertilization. What is ET? Yes, it is embryo transfer. What is ZIFT? Zygote intrafallopian transfer. What is IUT? Intrauterine transfer. Both are the type of ET. ET ke do type. Very good. Then some more are, what is GIFT? Gamete intrafallopian transfer. Okay. What is AI? Artificial insemination. Now these are the cases where the fertilization happens inside the body. Is there any chance of in vivo fertilization and embryo transfer? Yes, it is also required in some cases. And these embryos are not of the same person, but of a surrogate mother. These uterus is not of the same person, but of the surrogate mother. So embryo transfer is done in the surrogate mother. Okay? Right. In these two, there is in vivo. In vivo. Now the latest method of in vitro fertilization, the latest method is called ICSI. That is called intracytoplasmic sperm injection. Which cytoplasm? It is ooplasm actually, ooplasm. Intracytoplasmic, this cytoplasm is the cytoplasm of the egg or the ooplasm. Intracytoplasmic sperm injection. This is the latest method of IVF. Okay, so this is IVF only. AI, artificial insemination. Insemination. Now we can have one type of that is known as IUI. IUI simply means intrauterine insemination, or it can be done in vagina without any special abbreviation used here. So still use AI only. Okay. So these are the abbreviations. Once the abbreviations are understood, this topic is also over. Okay, bachcho? So we are almost at the completion of this part also. Principles of inheritance and variation. Approximately 4 plus 3, 7 questions. What you have heard and something that you have not heard, but you have heard earlier, okay? It is there in the mind. Have faith. It is there. Don't lose faith. Okay, so we are done with 7 questions out of 50. It may be more than that, but it cannot be lesser than this. Okay. Now next is we are doing for the 2 questions. Actually, this chapter is 3 to 4 questions, or sometimes 4 to 5 also. But generally 3 to 4 is this chapter. Major part of this chapter is in the botany, and the human genetics part is in the zoology. So we are expecting 2 questions, okay? 1 to 2, let us take it in this way, 1 to 2 questions in this part. So it is only yielding 1 to 2. Accordingly only we will give the time to it, as it is only giving 1 to 2. Now, sex determination methods, this is male heterogamety or female heterogamety. The male heterogamety is XX and XY method and XX and X0 method. And female heterogamety is ZW and ZZ and ZW and Z0, only one Z, okay? Heterogamete, male heterogamety, we are using XXXY or XXX0. Female heterogamety, we are using ZZ, ZW and ZZ and Z0. ZZ and Z0. Not ZW and Z0, I spoke wrong. ZZ and Z0. ZZ is the male and Z0 is the female. ZZ male, ZW is the female. Now examples of XX and X0 is grasshopper and most of the insect, in fact. Example of XXXY, human beings and Drosophila. Examples of ZZ and ZW, birds, most of the birds, hen, fowl, okay? And examples of ZZ and Z0 is a moth called Fumia, moth, or many butterflies also, moth or butterfly. So if they give moth or Fumia, generally they ask the question, so the example you have to remember is Fumia, Fumea, and that is of ZZ and Z0 method. Okay, Z0 means only one allosome. In place of 2, only one allosome. So number of chromosomes are 1 lesser. In male or female? In ZZ and Z0, 1 chromosome is lesser in male or female. Can you write male or female? By the time I can wipe. Female, very good. Varalakshmi. Very good. Chitrilekha, very good. Female. Random things. Okay. So today people are answering. So earlier what used to happen, if answers are coming, some are correct, some are wrong. Today, if answers are coming, they are only correct answers. Really good. Okay. That means at this pace also you are maintaining your understanding. It's a good thing. Beta sex determination, Hanking, he discovered the chromosomal method of sex determination, and first time he studied that in the insects, and most of the insects are XX and X0. Example, grasshopper, it's an insect, and it is also having XX and X0. So remember that the chromosomal method of sex determination was discovered in insects by Hanking, king of hen king. Okay, at that time he didn't call it as X chromosome. He called it as X body. He found that female egg has got two X bodies, and some egg has got some sperms. Okay, they have got either 1 or 0. Okay, the egg, where is the bottle? Yeah, the egg has got, all eggs have got one X body, and some sperms have got one X, and some sperms have got no X. That time he called them X body. Later on, the X body, what he called X body, was the something X. He didn't know that. That X body is called the X chromosome. They found it to be a chromosome later on, and then another chromosome discovered was the Y chromosome, and this is XX and XY method, which happens in the humans also. Humans and Drosophila. Give me a second. In which of the following examples the sex determination happens in the number of chromosomes? That means either male have got one extra or female have got one extra. I mean, male have got one lesser or female have got one lesser. So one lesser hoga to other ka extra automatically. Examples, okay? Examples are grasshopper, number 1, number 2, birds, number 3, Drosophila, number 4, butterfly, moth or butterfly. In this case, two examples are there, and that is the insect grasshoppers and butterfly, which is the moth fumia. Two examples are there where the difference in the number of chromosome is there. Okay, remember that in the birds, equal number allosomes are equal in both male and female. Very good. Now, in which of the following example the sex is determined by the number of sets of chromosome? Number of sets of chromosome in which of the following examples? Again, you have the birds, then you have the bees, then you have the insects, you have the butterfly. Birds, bees, insects, butterfly. Yes, correct answer is honeybees. Very good. The bees is the correct answer, and that is the number of sets of chromosome. Honeybees. Okay. So that's what birds and bees in our example. Okay, bachcho, then genetic disorders. So genetic disorders are of two types, Mendelian disorders. These are generally single gene disorder, single gene mutation, and they will follow the Mendelian pattern of inheritance. Therefore, they are called Mendelian disorders. Other one is the chromosomal disorders. So chromosomal disorders are chromosomal aberration. Aberration means either the number of chromosome is faulty or the structure of chromosome is faulty. Okay, either the structure or the number, or sometimes the both. That is called chromosomal disorders. Now, this usually happens due to aneuploidy, and hence we cannot determine it by pedigree analysis. So it is because it is not following the Mendelian pattern of inheritance, we cannot have the pedigree analysis available or possible in case of chromosomal disorders. Okay, beta. So Mendelian disorders, now it is a single gene mutation Now, whether this gene is present on autosomes or in allosomes, so accordingly we can have the allosomal disorders or autosomal disorders. Autosomals or allosomals, right? Autosomes, we have, the autosomes, and allosomes also, the sex chromosomes are called the allosomes. And all the remaining chromosomes are called the autosomes. Good. Autosomal disorders or allosomal disorders. In allosomal disorders, the disorder can be X allosomal, Y allosome, X linked or Y linked. Generally, what we have in our examples are X linked only. We don't have any Y linked example of any disorder. Generally, Y linked traits are there, disorders are very few. Y linked traits are there. So, whenever we say allosomal, we are actually meaning the X linked one, sex linked one. But, okay, so autosomal, now it is a single gene mutation, whether the mutation autosomal, or it is allosomal. In allosomal, it has got either the X-linked or Y-linked. We are actually only remembering, or we are only considering the X-linked disorders. Okay. Now, whether this gene mutation which has happened is a dominant mutation or it is a recessive mutation. As you know that most of the mutations are recessive, so most of these disorders names are also in this category. So, autosomal and recessive, and autosomal and dominant. Similarly, X-linked also, X-linked recessive and X-linked dominant. Now, these are the Mendelian disorders. Mendelian disorders are single gene mutation, the gene present in autosomes. So, these disorders are called autosomal. If the gene present on the allosome, X-linked genes or Y-linked, this is called allosomal. Okay, all these are the Mendelian or genetic disorders. Okay, good. Now, examples of the recessive one. Examples. So we have got some very important example like sickle cell anemia. We have examples like thalassemia, sickle cell anemia. We have examples like phenylketonuria, PKU, phenylketonuria. We have examples like cystic fibrosis, albinism, sickle cell anemia, thalassemia, phenylketonuria, cystic fibrosis, albinism. Now, these 5 names are either described in this chapter or they are used somewhere or the other. Cystic fibrosis is used in some examples of the, genetically modified animals. Albinism. So you may get these names these names are there in the NCERT. Now, dominant, we have only one example that is called myotonic dystrophy, myotonic dystrophy. And in case of X-linked and recessive, so 2 very important examples is color blindness and hemophilia. The X-linked and dominant. Now, X-linked dominant, rare, but one of them is called IP, incontinentia pigmenti. IP, incontinentia pigmenti. I told you that most of the mutations are recessive, so most of our examples are in the recessive category. As number of autosomes are more, so therefore the autosomal recessive examples are also more. Okay, so therefore in terms of probability of the disorder, so there are more chances of autosomal recessive disorder. So why? Because autosomes number is more, allosome is just an X-linked one, and of course recessive, most of the mutations are recessive type. The difference of recessive and dominant is just when single copy of the gene is sufficient to cause the defect in the body or the phenotypic defect, then it is called dominant mutation. But if a single gene, if we have got out of the two alleles of the gene, if one allele is normal and one allele is affected, then the normal one is actually taking care of the phenotype and not any phenotypic difference or defect happens. Okay, so to have the defect, you need two such faulty genes. So both the alleles, okay, should be faulty from both the parents. So that is, and this is the reason that why the mutational disorders are rare. Why they are rare? Okay, number one, the survival is less, the viability is less. Okay, so people are not born with, the embryo cannot sustain. If at all they are born, okay, so recessive, because most of the mutations are recessive. So people who have only one faulty gene, they are not the diseased person, they are just the carriers. But so please remember that in case of dominant, there is no concept of carriers. Dominant में carriers ले दूं, dominant लो carriers ले दूं. Okay, carriers are only in case of recessive. Good. So the other example of the chromosomal one are due to chromosomal aberration. Aberration is either defect in the number of chromosome or defect in the structure of chromosome. Either the number of chromosome, when the defect is in the number of the chromosome, generally we call them as aneuploidies. Okay, in animal we have aneuploidies, but we don't have euploidies here. We are only having aneuploidies. And aneuploidy is 2n plus 1 or 2n minus 1. Okay, as the definition goes, it can be 2n plus 1, 2, 3, anything. But we don't have any viability if there are more than one chromosome. So either one chromosome extra or one chromosome lesser. In this case, total number of chromosomes are 47, and in this case, total number of chromosomes are 45. Okay, Vicha? Then we have got the structural one. Structural one are deletion, duplication, then translocation. These are the structural. Defect in the structure. Defect in the structure of chromosome. Defect in the structure of chromosome. Examples are deletion, duplication, translocation, which can be a type reciprocal translocation also. A type of blood cancer happens by reciprocal translocation. Okay, deletion, there is a disease known as 5p minus disease or Cradock syndrome. Okay, then we have got these aneuploidies. The reason for aneuploidies are ND. ND stands for non-disjunction. Because of the non-disjunction means failure of anaphase. It means failure of anaphase. Either anaphase first or failure of anaphase second. Either failure of separation. So anaphase में defect होगा, failure of either in the case of first primary non-disjunction or in the case of second one, secondary. Failure does not mean that it will not happen. It will happen, but then some chromosome are not actually separating. So better word here is defect in the anaphase. Non-disjunction, okay? Disjunction happen at the anaphase, and non-disjunction, that means the separation is not happening. Okay, beta? So examples of 2n plus 1. So 2n plus 1 example, one of them is known as the Down syndrome. Okay, then you also have allosomal aneuploidy. It is autosomal aneuploidy, because in Down syndrome, chromosome number 21, there is a trisomy 3. In place of 2, all of us have 2, but the defect or the patient of Down syndrome, he has got 3, chromosome number 21. Ante in all of us, out of the 2 chromosome number 21, one has come from Nana and one has come from Amma, one from father, one from mother. But in the case of the Down syndrome, what has happened? Either 2 have come from the father or 2 have come from the mother. Okay? Either 1 extra from the father or 1 extra from the mother. All of us have got only one. Down syndrome. Then we have got the aneuploidy, that is in the allosomal aneuploidy. Allosomal aneuploidy, we have examples like Klinefelter's. Klinefelter syndrome, it is XXY. So male with extra X chromosome. A male with an extra X chromosome, that is the Klinefelter. And 2n-1 example, that is the monosomy. If this is called trisomy, both are examples of trisomy, Down and Klinefelter. Down in autosome and Klinefelter in the sex chromosome or allosomal. And 2n-1 therefore is called monosomy. Monosomy matlab either of the chromosome, koi bhi chromosome, in place of 2, if this person has got only 1. Means that either from the egg there is 1 less, or in the sperm there is 1 less. Okay, that is example. So example of the monosomy, we have got the one example of monosomy, that is called Turner syndrome. So Turner syndrome is a female with a single X chromosome. A male with an extra X chromosome is Klinefelter, and a female with 1 lesser X chromosome, that is called Turner. But you remember that in all these examples, deletion, duplication, translocation, or reciprocal translocation, aneuploidy, 2n+1, 2n-1, that means trisomy or monosomy. In all these examples, we cannot have the defect will run in the family, just like the Mendelian pattern of inheritance, and therefore the analysis in the pedigree is not possible. So इनका कोई भी pedigree analysis नहीं होता. Please remember that Klinefelter or the Turner, the both of them have one thing is common, and one thing is common is both are sterile. Both are sterile, not infertile. They are sterile, totally sterile. ये gametes ही नहीं बना सकते. They are sterile, okay? Because for gamete formation, the completion of the meiosis is imperative. It is important, and for meiosis, you should have the prophase first, and prophase first has got the zygotine. Zygotine means the pairing, and here the problem happens of pairing only. So because pairing cannot happen in them, and therefore the meiosis is not possible, and because the meiosis is not possible, the gametes are not formed. And if the gametes are not formed, that means they are sterile. So what is common to both of them is sterile, but in one, it is what is different in them? One is a male, one is a female, one is a trisomy. Which one is trisomy? Klinefelter. And which one is monosomy? Turner. Remember that. Monosomy के example हमारे पास only 1. Trisomy के examples 2 हैं, Down syndrome and Klinefelter syndrome. But if you can really have faith in me, don't take tension about the pedigree analysis. If at all any question comes, okay, at that time only you can analyze it. We have seen last 10 years. Very few questions, we could only locate 2 to 3 of them. So pedigree analysis. No doubt, when we are doing the chapter in the classes, we are doing that. And when we are giving 50 questions from one topic, certainly we are giving more than 1 or 2, 3 questions we are giving on pedigree analysis, okay? But it is not that important topic for the final NEET. I'm not saying it will not come at all. And even if it comes, okay, the diagrams of the pedigree of the NCERT textbook. They will simply pick it up and give that, and you can identify that. You can easily identify that, right? So pedigree analysis is not a very important one. It is interesting one, right, in terms of genetics, but then it is not very important for the NEET UG examination. So right now, if we do the pedigree, it is really cannot be done in this class. Now, अभी शुरू हो गए बेटा. Skip generation are common in X-linked. Yes, Lakshmi, you are right. Okay, in case of X-linked. Okay, good. In case of recessive only. In case of recessive, whether X-linked or autosomal, recessive means skipping of generation happens in recessive. Okay, I cannot understand these Telugu language, so therefore people are writing that. That is for sure. They are not talking in terms of biology or the subject. Okay, skip generation is common in recessive disorder. Very good. You are right. Chinmay, you are right. Okay. Okay. Right. Questions. Anish Arya. Questions. Better. There are all type of people are there. Some type of people are there. So you have given so many questions. Okay. We are doing the questions daily, but we have not seen the NCERT. Okay. So we have not seen the NCERT for the last 2 months. If you can give a quick revision of the NCERT, that is good. I also found that although I'm doing, so whatever I am discussing right now, if any word which I am saying, it is actually the questions only. I am just giving the explanation of the questions. I am only telling whatever the questions, their explanation. Because I can easily show you how we can make whatever I am saying in these 5 hours. We can give you 200 questions out of that, easily. Okay, so obviously 50 questions are very, very easily we can give from that portion. And when you see the question, you will find that, yes, sir, you spoke about this. Yes, you spoke about that also. So only those things we are doing. Okay, done. Now, bachcho, the second portion of the genetics is the Human Genome Project and the DNA fingerprinting. Okay, let us open the NCERT book and try to understand that. Here also many people have doubt. No doubt about that. The techniques are really confusing, but please understand that the techniques are actually, so these are the pedigrees which are given. Okay, the pedigree symbols can be given, or the pedigree diagrams, only these 2 diagrams are given. The maximum that can happen, that can come from the pedigree analysis. Okay, done. Molecular basis. ये वाला portion though, this portion up to here is the botany portion. So I am directly coming to the Human Genome Project. So it is launched in the year 1990, completed in the year 2003. All these data you already know. The vectors used are BAC and YAC. That also you know. Okay. And equipment uses automated DNA sequencer, ADS. Automated DNA sequencer is based on Sanger's method. Now, Sanger was the scientist who got the Nobel Prize for finding out the insulin structure. So all the amino acid sequence in the proper, the 2-chain, chain A, chain B, and also the chain C, that was given by Sanger. For that, he got the Nobel Prize. Okay. And on that basis only, on that same technique, they have developed the automated DNA sequencer, wherein the nucleotide basis sequence can also be determined. Just like the amino acid sequence, here we are doing the nucleotide sequence. Coordinated by the US Department of Energy and NIH, National Institute of Health. Please understand that this NIH is not the Indian one, it is an American one, NIH, USA. The contributors are later on the Wellcome Trust UK, they become the partner, and after that many companies, small or big, many pharmaceutical firms and many institutions, right, they have become the partner, including in India we have got that Bangalore wala, one they also become the partner. In Bangalore we have one big institute of sciences, Indian Institute of Sciences, okay, I think like that. So that has become the major partner, okay. And similarly, similar type of high repute institutes from Japan, from France, Germany, China, and other countries, okay. The goals were simple, we all know. Now comes the methodologies. So the two methodologies are EST method, wherein we are making the EST. That is, EST means the gene. Okay, so genes are actually made into, the genes are made, and the genes are made from the expressed RNA. So the mRNA, from the expressed RNA, the genes are made. Now you know that how we can make the genes from the RNA? We can make the genes from the RNA or DNA from the RNA by reverse transcription. And you need not to do by yourself. You have got many viruses. You can hire them and they can do it for you. Okay. So they can convert this RNA into the DNA. They have those enzymes to do that. Very good. And so we can make those genes and we can analyze those genes. But then scientists call, no. When we have enough of money and so many people are available, so much of expertise available, why only do the express sequence? Because bachcho, less than 2% is expressed. Less than 2% is expressed as mRNA. So they are saying, no, this is not right. Less than 2% is expressed as mRNA. Right. So later on they decided for Human Genome Project, they have done the sequence annotation method. That means they have done the blind approach. Blind approach matlab complete sequencing was done. It is called the blind approach. Not seeing that whether it is expressed or non-expressed, it is exon or intron, not seeing that. It is called blind approach. Sequencing the whole set of genome that contained all the coding and non-coding sequences. Later on, they can identify which one is the coding part, which one is the expressed part, and which one is non-expressing or non-coding part. They found that less than 2% was the coding part. And coding part, it makes the mRNA and ultimately the proteins. Okay, so that is the genes, the genes, the genomic part, the genes. Total number of genes that were identified are how many? 30K. 30K genes were identified. That was the features of the human genome. Now, bacho, I think features of the human genome, just by a single look, you can recall whichever is not clear or whichever you are forgetting, you can simply recall it. Okay, so don't think that you have any problem or any weakness in Human Genome Project, because this is only the memory part. How many? What is the size of the genome? The size of the genome is 3164.7. Okay, or it is called 3164.7 million base pairs, or we can say 3.1647 billion base pairs. So please see where is this decimal. It is after the 3, then you can call it as billion. If it is after the 4, between 4 and 7, it is the million. 3,000 million or 3 billion, like that you can remember. Average gene consists of 33,000 bases or base pairs. The largest gene is dystrophin with 2.4 million bases. 1.4 million are the SNPs or single nucleotide polymorphism. Okay, these arise due to mutations. And then comes the DNA fingerprinting. DNA fingerprinting is the shortcut method of identifying someone from the DNA, Right now, identification is done by seeing the face, but then face can be deceptive. So how to rely? We can rely on the fingerprint of the real fingers. That is also called dactylography. But the main problem comes with these fingerprints is that you can get them changed. A simple surgery, they can do a small surgery, you can pay 10,000 rupees and they will change your fingerprints. And if a small change happens, the machine will not read as the same print. So therefore, people can save themselves. But then if they have done DNA, if they are using the DNA and they are identifying a person, they are doing DNA fingerprinting, that cannot be changed ever. So DNA is never deceptive, it is always truthful. So DNA fingerprinting was developed by Alec Jeffreys. Remember the pioneer name? Many a times it is asked. And it involved identifying differences in some specific regions in DNA sequence called as repetitive DNA. This is 2022 direct question. Same. And the answer was repetitive DNA. What is bold here was the answer of that question. It involves, so the question was DNA fingerprinting involved identifying differences in some specific regions in DNA sequences called as, okay, and the option were many, and one of the option was satellite DNA also. But then repetitive DNA was the answer. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. So when you are using an ultracentrifuge machine, you can separate them. And how you can separate them? Ultrafuge machine, you have to put the sample in this test tube and then ultrafuged, okay? Ultracentrifuged. Okay, now this already contains one solution there, and that is the hypo, cadmium. What is that? Okay, so here there is actually a chemical is already there, and that is having differences in the density. That means initially the density is lesser, but as you are going deeper and deeper, the density is increasing. And very carefully over that only you have to pour your DNA sample, and then at that time it will not mix up. Later on, you have to put this test tube in the ultracentrifuge, and after the completion of the centrifugation, then you can take it out, and then you can find that the satellite DNA has been separated as different bands, but you cannot see them directly because it is all transparent only. So what you can do, now you can use a machine which can identify in terms of what happens to the light, okay? So, but still we cannot notice that by our eyes only. So we need a photometry method to identify that. So photometry method of this test tube containing different bands of different density, okay? When we do that, then we get this type of result. This major peak, this is of high density. So high density is usually the genomic DNA. This is the coding region. This is the major does not mean that major in terms of quantity. No. This is only density. Major peak, bigger density, higher peak. Okay. So this is not to be used for DNA fingerprinting, because this chinna peaks near the big peak, chinna peaks are called the satellite. Planet, chinna satellite. So planet peaks and satellite peaks, okay? So satellite peaks some are called, the mini satellite, some are called the micro satellite, mini satellite, micro satellite. So major peak, minor peaks, in fact, minor peaks are some more than one type. So some of them are mini and some of them are categorized as micro. Minor peak, it comprise of repetitive DNA or satellite DNA. Now, why I said that this question came, actually the teacher has given in the main NEET examination repetitive DNA as well as satellite DNA, and I am skeptical that answer could be both. Okay, because repetitive DNA and satellite DNA is only, it comprises of the repetitive one. Okay, but the answer was given as ditto as of the NCERT line, and no one has claimed also. Why? Because when it is given in the NCERT line, word by word, same line, then no one has really claimed it. But no doubt the satellite DNA as well as repetitive, both options were there. You can see the question. This question came in 2022. Okay, how you can cut the DNA? We can cut the DNA into small pieces by using restriction enzymes. After that, we can separate the satellite part. In the satellite part, we have got the mini satellite and micro satellite. Now, depending upon which method is being used in your laboratory, for example, Alec Jeffreys were using the mini satellites. Okay, so the mini satellites, a type of mini satellite, he called them as VNTR. VNTR belong to mini satellite. VNTR, variable number tandem repeats. Variable number tandem repeats. Okay, some latest laboratories are not using the VNTR because they are not taking from the mini. They are taking from the microsatellite, and they are calling it as STR, short tandem repeat. Short tandem repeat. In the diagram of your NCERT textbook, they have actually used this word short tandem repeat. Okay, so number of STRs, number of short tandem repeats. So now again come, where is VNTR? VNTR will be found here. So if Alec Jeffrey has to take the sample, he will take the sample from this portion. And today in most of the laboratories, including the laboratory in India, in the Hyderabad, they are taking it from this, the microsatellite. Okay. Also remember, the microsatellite only is used today for doing the mapping of the genome. So mapping of the genome, they are using the microsatellite, not the mini, microsatellite. Microsatellite, because microsatellite DNA is different in all of us. So this is showing polymorphism, including this also. VNTR also show polymorphism and STR also show polymorphism. But STR is easier, it is more conclusive, and also it remains conserved in a long-standing sample. So even if you take a sample after a few years also, then also the DNA has not degraded. Okay, but then the VNTR, we have seen that after many years, the VNTR start breaking, and then you cannot get the right result of the long-standing samples. Okay, therefore today they are relying on the microset. How they are different? They are just different in three categories. They are different in number of the first of all, the size. How many base pairs are there? That means the size. Okay. Then how many repeats are there? The copy number. And number three, whether it is having more of CG or more of AT, like that. So that on that basis, the two micro or mini are being differentiated. The steps I call them as IDEB, IDEB, and HA. Okay, HA, IDEB, ha, I call them. Why I do ha? Because of repetition. We have to always locate the repetitive part, and the steps include the isolation. So you have to isolate the DNA from the sample. The sample can be the blood sample. In the blood, we are not using the erythrocyte, obviously, because they don't have the nucleus. Okay, or even any other DNA is not there at all in the erythrocyte, but in the leukocyte we can find out the DNA. Okay, done. So we can isolate from the sample. Isolation. I'll write first of all IDEB, and I call them as ha, repetition, and I just ignore many repetition. I just focus on this word. Okay. So I then stand for the digestion. Digestion matlab hydrolysis, but that means breaking by using the endonuclease, restriction endonuclease. We can use them and then we can cut from the precise region. Okay, we have got more than 3,000 type of RE today, and we can use the precise one, whichever your VNTR you are using. According to your VNTR, you can use the RE. Okay. Now after that, we have got the E. E stand for electrophoresis. Electrophoresis. This is gel electrophoresis. Okay, gel electrophoresis. Then B stands for blotting. Blotting. This is called southern blotting. Whenever you are doing the blotting of the DNA, it is called southern blotting. And blotting is done on the nylon membrane, because after the blotting, the DNA will not move from the nylon membrane. Okay, it will remain there, and later on you can also document it. You can also document it. You can of course document the printout also, but you cannot document the gel, because gel, if you keep for a few hours, it will move and you will not find the DNA in the right place. So you have to immediately transfer it on the nylon membrane, and when you are transferring it, this technique is called blotting, southern blotting to be more precise. And HA, and H stands for hybridization. Now hybridization is done after when we untangle the two, or by using some alkali, we can denature the DNA. Denaturation में क्या हुआ? इनके बीच के hydrogen bonds breaks up and it become untangled, it becomes single stranded, and now it is ready to hybridize with the VNTR probe. So whichever VNTR you are using, of that probe you can use that VNTR probe, and when VNTR probe is allowed to hybridize with any of these DNA, so we call it as hybridization. After hybridization, then we have to do you have to take the printout on the photographic membrane, photographic plate, and that is called autoradiography. Now, this probe, VNTR probe that we are using, the probe comprise of the, we can call them as the DNA, single-stranded DNA. We of course also probe, genetic probe are also RNA also, but here because we are identifying the DNA, so we are using only the DNA probe. So DNA probe is a single-stranded one which is labeled. Now labeling, in this case, labeling is done with radioisotopes. Now why radioisotope? Because if you keep them in dark, and of course you can use one photographic plate, you can have some impression by autoradiography. This is called auto, because from the cells, from the DNA only radiations are coming. This is called autoradiography. And graphy means to write. That means you can write from the lines that are rays coming out from the DNA, the autoradiography. Okay. So these are the steps: isolation, digestion, electrophoresis, blotting, after that hybridization and autoradiography. But then if you can add one more process, then this can become very much sensitive. Sensitive matlab, then chances of errors are less. If you add just one more technique, so what is that technique? If you add in any of these, in any of the place, if you add this technique, it can become more sensitive. Chances of faulty answers are lesser. Chances of faults are less. This is called more sensitive. What we can add here? Tell me, bachcho, what we can add. Meanwhile, I can take a small breath here. Water. Yes, you're right. You're right, Sudheen. It is PCR. Very good. Sudheen, Brighty, PCR. If we just add PCR, it can increase the sensitivity of DNA fingerprinting. Now, those who are revising this topic, I always say, don't revise from the chapter, please revise from the questions. And try to find the answer of the question in the chapter. Retrograde. Because if you try to do from anterograde, the regular method, we are reading the NCERT, you will read it 20 times. Still, you cannot get the entire technique, because no doubt we are not, neither we are showing you the technique nor you are doing it in the laboratory. Okay, so PCR polymerase chain reaction. Yes, you are right. Correct answer. One more question. At which stage we can use this PCR? You can say, sir, before isolation or before digestion, sir, before electrophoresis, before blotting, before hybridization, or before autoradiography. At which stage, if I add PCR, adding PCR means just adding one more technique. You can take the fragments and you can multiply them. You can multiply them, you can amplify them. So at which stage we can do that, Bacho? Before what? After blotting. I'm sorry, beta, not a correct answer. Narayan Sai. Early, early kab, beta? Before hybridization? No, sorry. Before digestion? No. Yes, before separation. How we are separating it? What we are using? We are using the age and page method. Yes. Before electrophoresis. Here, PCR, if we can, because after digestion, you will have DNA of different sizes, no? You will have DNA of different size, small, bigger, different size. This is a variation, this is a polymorphism that we are talking about, and this only we have to see by the further techniques. We have to identify them, we have to see them. For that, we have to separate them. That what happens that you have to use the electrophoresis method where gel is being used on a microtiter plate. What happens? Some small fragments, if they are fewer in number, then chances are there that they are not blotted properly, and if they are not blotted properly, they are lost, and therefore your answer will be having lesser fragments. So chances of errors are more or sensitivity is less. But if you use the PCR, you can amplify all of them. So many thousand times you can amplify them, and after that you can use the electrophoresis. Don't worry that if a same size, maybe 1 or maybe 100, all will be separated at the same place only. This is the process, specialty of the process called gel electrophoresis. The separation is done because the DNA is negatively charged, and therefore it is moving towards a positive electrode, and once it is attracted, the smaller fragments, they can move further, and the bigger fragments, they remain closer to the baseline or closer to the well region. Okay, so I hope you understand. So before electrophoresis, after digestion, before electrophoresis, if we do that, then we can get a better result. The first feedback, the first review, I want you to give the feedback. Just tell me what we can, because you are giving time, I am also giving time. Okay, we can utilize the time better. We have heterogeneous, the other end, heterogeneous crowd. So majority of the people, we can follow their instincts. Okay, so now we have I have almost completed 11, the minimum I have completed 11 questions. Okay, so Tinara, so early, 10:33 only. Evolution, yes. No. My point here is the way we are doing, or we can take some more time to explain some topics, or no, sir, we are just doing, because if we see it, we can recall many things. And if we have not remembered is something, this is not the time to, because even if you remember now, you cannot recall in that pressure time. The final examination is a pressure examination. In that that constraint of time, pressure, performance, only one exam, full life depends on one. All these pressures are there. What happens that you are only playing with your reflex. You are not doing any technique or identifying which is out swinger or in swinger, or it is coming from this type of hand or this type of hand. No, no. You are only playing with your instincts, with your reflexes. Okay. So reflexes means that anything you try to remember now, nahi yaad aayega beta. You can remember in that time, nahi yaad aayega. Generally, this is the time to reinforce, okay, consolidify whatever already you have done. Shall we move? Why you put the vlogs? A quote is there, then love sign is there, and paper sign is there. What is that? Tell me. थोड़ा बताओ बेटा क्या है ये? Okay, beta. Okay, Sudhin giving such a good one. बता दो बेटा क्या है ये? Coat. Okay. Okay, apron. Okay, apron, love, test. Love from. क्या आ गया? थोड़ा और ऊपर जाए. Okay. Anyways, good. ये बी से क्या लिखा है? Love from Bihar. Okay, anyways, from now just review briefly all the important topics. Small chapters, we can do them. This is the right thing. And yes, beta. Evolution. Now, this is a chapter where explanation at this time is really not required. Even if it is required, then if you try to remember it now, just recall so that you have confidence to answer. You have confidence to answer all that. Okay, so right. Here, all these brief things, okay. 4.5 billion years is the age of this Earth, age of the solar system also. And about 4 billion years ago, life in any form has come, okay? But then when we can say that the non-cellular forms of life, those forms have come about 3,000 million years ago only. And the cells, the first cell, the prokaryotic cell, the cellular form of life has only come 2 billion years ago, okay? So like that, if questions will be asked. Now, this is the diagram being, the experiment being done by Louis Pasteur, and he is refuting the concept of abiogenesis. He is giving the concept of biogenesis. Biogenesis matlab life comes from the pre-existing life only. But no doubt it is not explaining how the first life come. New life come from the pre-existing, but how the first life come, this is not giving the answer. Anyways, so chemical evolution given by Oparin and Haldane has tried to explain how the life has come. So life has come by series of chemical evolution. Then we can get the first life form, and from the first life form, all what happens is the biological evolution. Chemical evolution is over at that time, biological evolution is continuing. Okay, but then first time is abiogenesis, no doubt about that. The first life came from the chemicals only. But yes, that is only possible at that time. The conditions were different. The conditions were very high temperature, no UV cover, no ultraviolet. So then, no ozone cover, sorry, the ultraviolet rays are coming down, and that is causing a lot of reactions to happen spontaneously. High temperature, again, it's also causing lot of volcanic eruptions, and all these radioactivity was high. All these things are, so conditions are different, but the biggest factor is that time it is reducing atmosphere, because in an oxidizing atmosphere as of now matlab, although we have only 21% oxygen, but when we say oxidizing, oxygen exists in O2 form, molecular oxygen. Okay, done. So the reducing atmosphere, and in a reducing atmosphere, the problem of self or auto oxidation are not there. So chemical evolution could be possible. Okay, bachcho, evidences of evolution, paleontological evidences, these are the evidences from the fossils. Then evidences from comparative anatomy and comparative morphology. So when we compare the anatomies and morphology, we find some homology or analogy. Homology in terms of organ is homologous organs and analogous organs. Now we know that homologous organs, they are indicating common ancestor. Analogous organs are indicating different ancestry, but similar, similar changes, and similar changes, similar functions. Okay, why? Because the environmental challenge is same, and therefore the same adaptive changes have happened. Now, this diagram has been asked earlier. You can find this diagram in the PYQ books. Okay, so this is showing the homologous organs in the plants. So thorns and tendrils, thorns of bougainvillea and tendrils of cucurbita, sorakai, cucurbita, and bougainvillea, very common in our state. Okay, now this state is my state, but my where I was living earlier. Okay, thorns are axillary buds, and tendrils are also axillary buds. Okay, bachcho. So then forelimb of mammals, hind limb of mammals, all these are also examples of homologous organs. Forelimbs of mammals, hind limb of mammals, these are all homologous organs. Okay. Molecular homology means the similarity in the molecules. Okay, or it is also called biomolecular homology. Now, in this case, you will find that similar molecule, similar protein, similar hormone in diverse organisms, similar hormone, similar, even the genetic code is same. ATP, energy currency, same in all the life forms, right? This is molecular homology. Whenever the homology is being studied, always remember, homology is indicating common ancestry. Common ancestry, that means that now divergence has happened. So this type of evolution, which indicates common ancestry, but now distinct or diverse type of organisms, but from common ancestor. This type of evolution has been called as divergent evolution, and opposite to this effect is a convergent evolution, when we can notice some analogy. Okay, so embryological, this is, I don't know why people are having doubt in embryological evidences. Embryological evidences has been so much goofed up in NCRT that now this concept is so much of treated so well in the textbook. No need to take tension. वो तो examiner के tension है. It is our tension to give questions, not your tension. So what question we can give you? Who gave the concept of, this? Although this ontogeny recapitulates phylogeny, ये भी नहीं है अब तो. Who has given the embryological evidences only that you can remember? Embryological evidences were given by Ernst Haeckel. ठीक है. Who has disproved, or who was not happy with the embryological evidences? Von Baer. What Ernst Haeckel was saying, Unethical was saying that the embryos contain some structures, or these are non-functional but they are still existing in the embryos, and they are not present in the adult life. So why they are in the embryos, and they are found in the embryos of many distinct forms. This shows that all of them have a common ancestor, right? So this is what the this is showing. Or this is ontogeny recapitulates phylogeny, or in case of that, I mean, like how we can deduce. For example, if the embryo of frog contains gills, adult frog does not contain gills, but the embryo, the tadpole, contains gills. So who has got the gills right now? The fish have gills. That means frogs have been evolved from fish-like ancestors. Now, this type of ancestry we can understand from them. Similarly, if all the embryos of we can say amphibians, reptiles, birds, and mammals, all of our embryos have got gills, non-functional gills, but they were there, adult ledu. That means we all have come from guild ancestor. हम सबका ancestor common guild ancestor था. This is embryological evidence. One buyer said embryological evidence. He said that embryos never resemble or never pass through the adult stage of any ancestor. So he was not happy with that. Just like that. No need to just remember the name, who gave, who refuted. Adaptive radiation. It is a type of microevolution wherein many different species are formed from a common species in an isolated geographical region which contains many different type of habitat. Okay. Now, in an isolated geographical region, like in case of islands, you will find a lot of adaptive radiation. Now, one such, in fact, a big island is Australia. Australia got separated from the mainland, as you all know that. It is separated from the Eurasia, and that is, it had become a distinct island. Okay, in Australia, different type of habitats were there, and therefore all the mammals there, we can find that they have been diversified into different forms. Okay, so if you find any marsupial or the original marsupial, today we have got different type of marsupial, and this developed by adaptive radiation. So it has occurred in an isolated geographical region representing many different habitat, and this has resulted into formation of different species. So by adaptive radiation, you can only find a species or subspecies, but you will not get bigger taxonomic group. New phyla will not develop. New classes will not develop. Class, phylum, they have developed very long time of evolution. It is a macroevolution. So then we don't use the word adaptive radiation. Then it is better call it as divergent evolution. So please understand that all adaptive radiation are actually divergent. They are actually divergent evolution. But of course, in divergent evolution, it is a longer time frame, and it has given rise to many different taxonomic groups other than the species and subspecies. Higher taxonomic group, it takes a longer time. Okay, so adaptive radiation, one more example of adaptive radiation is in the case of the Darwin finches. Human beings, we don't show any example of adaptive radiation because today we have only one species of humans surviving, Homo sapiens sapien. And if adaptive radiation would have occurred, today we will have many different type of man. One man from Tasmania, different type. One man from that island, different man. One man, and different men are there. But today we don't have cows. We have different races, but all belong to the same species. Okay, bachcho? This is adaptive radiation. Not only the adaptive radiation of marsupial, in Australia, we have got adaptive radiation of the placental mammals also. So many different type of placental mammals have developed in Australia. For example, the mole of Australia, the anteater of Australia, mouse of Australia, the lemur is only of Australia. Okay, the flying squirrel, bobcat, and wolf, these are of Australia. Now these are the placentals. placentals. Now you'll find that there even the marsupials. Marsupial matlab pouched mammals. Pouched mammals. Placental means they have got a firm placenta. So the further development does not occur in the pouch. And these are the marsupials. Example of marsupial from a common ancestor. These examples are marsupial mole, numbat, marsupial mouse. These are all examples. Okay. But then what has been found that in the same place, if in the same place one mole is diversifying and this mole from the placental or mole from the marsupial, they will have almost the same qualities. Almost the same qualities, the same structures, the same behavior. Reason being, they show convergence. Why they show convergence? Why? Because they belong to two different groups. One is a marsupial, one is a placental. But why we say convergence? Because they are showing similarities, right? They are saying, so when more than one adaptive radiation appear to have occurred in an isolated geographical region, okay, when more than one adaptive radiation appeared to have occurred in an isolated geographical region representing many different type of habitat, then we call this adaptive radiation as convergent evolution. Only this one, okay? So convergent evolution किसमें show करेगा? Mole and marsupial mole, anteater and numbat, mouse and marsupial mouse, leber and bobcat. In this we show convergent evolution, okay? But then what type of evolution do you find between anteater, lemur, mouse, mole, bobcat, okay, flying squirrel? This is all adaptive radiation. That means divergence. So one divergence and another divergence, if both are happening at the same place, they are showing convergence, convergent evolution. So this is special quality, and please remember this example, these examples of that, and also the statement when more than one adaptive radiation appeared to have occurred in an isolated geographical region, one can call that as convergent evolution. Evolution of life form, we have Lamarck theory. Lamarck was a French naturalist, and Darwin's theory. Darwin's theory is a theory of natural selection, and another person who has given the same theory is known as the another person who has given the theory of natural selection. क्या नाम था उसका? Alfred Wallace. Alfred Wallace. Good? ठीक है बेटा? So Lamarck theory, the two main points of Lamarck theory are inheritance of acquired characters and use and disuse. And the two main points of the Darwin's theory are branching descent and natural selection. Okay, branching descent and natural selection. These are the two main points of the Darwin's theory and the two main points of the Lamarck's theory. Inheritance of acquired character and use and disuse of organs. Natural selection is a process in which heritable variation enabling better survival are enabled to reproduce and leave greater number of progeny. Okay, the selected being are just they are selected, they are having more fitness. Now how they are fit, how they are more fit? Do they have a flat tummy? No, more fitness. This is the evolutionary fitness. Evolutionary fitness is only reproductive fitness. They are reproductively more fit. Means what? They can reproduce more. They can produce more offspring. Out of 4, if one can produce more than the other 3, then that means that this one is showing reproductive fitness or evolutionary fitness. Fitness. It means simply means that who can give more number of offspring in simple language. Okay. Now the points or the highlights of Darwinism. The highlights of Darwinism, theoretically populations. So these are based on some points, and some of them are factual. And these points are the factual points are these. Theoretically, population size grow exponentially if everyone reproduce maximally. But fact is, population size in reality are limited. But if you make a graph, you can find it, growing in geometric progression. That means multiples, 1 become 2, 2 become 4. But in nature, the size is not changing. Okay, seasonal fluctuations happen. But if man is not there, you will not find that after 10 years the number of giraffes come down or this number of species become extinct. Okay, so this okay, so you'll find that the population size are more or less constant. Now, don't think that, sir, is it true? It is not true, because even before man, many extinctions happened. Beta, ये Darwinism के highlights हैं. Darwinism, okay? So please understand that. So as per Darwinism, we can say that theoretically population size grow exponentially if everybody reproduce maximally, but fact is that population size in reality are limited. Natural resources are limited and population are stable in size means that there had been competition for resources. Whenever competition is there is struggle for existence. Members of population are not same. They are different. They vary in characteristic. Even though they look superficially similar, they have got different phenotypes. Those characteristics which enable some to survive better in natural conditions, which are the given climatic condition, given availability of food, physical factors, other, etc., would outbreed others that are less endowed to survive under such natural conditions. Okay, so those who are better adapted, they are more fit, they can produce more offspring. And how they are more fit? Because they have characters, they have the variations which can allow them to really sustain in that special type of condition. Okay, variations which are heritable and which make resource utilization better for few will enable only those to reproduce and leave more progeny. Please understand that in evolution, when we talk about variation, we are only talking about heritable variations. Heritable, that means all these variations must have a genetic basis. That means that it can be passed on to the next generation. Okay, so if any variation which is allowing them to use the resources maximum than others, for example, if a variation in one species to graze or browse easily than the other, obviously it will reproduce more. We can say it has outbred the others. It has outbred the others. Those who are better fit will be selected by nature. Fitness is the end result of the ability to adapt and get selected by nature. Okay, the fitness kaise aaya? Fitness aaya because of this ability to adapt and get selected by nature. The fitness, according to Darwin, refers to the reproductive fitness. Fitness, according to the gym instructor, is the body fitness. But fitness, according to Darwin, is the reproductive fitness. Another scientist who has given this theory is Alfred Wallace. He did not work along with Darwin, but he worked in Malaya Archipelago. Malaya Archipelago means today we call it Indonesia. Came to similar conclusion as of Darwin around the same time. In fact, to be more precise, little earlier than Darwin. Mutation theory. Mutation theory proposed by Hugo de Vries. He worked on evening primrose or Enothera lamarckiana. Evolution is caused by mutation, that is the point of the mutation theory, and not by minor variation. He says that one big variation, one big mutation has given rise to a new species. Okay, this is as per the Hugo de Vries, or the mutation theory pioneer. He says that one single step, a large scale mutation will happen. It will take a big jump, a high jump, or he call it as saltation. Saltation means leaping. Saltation, a single step, large scale mutation or saltation. This can give rise to a new species, and giving rise to new species is called speciation. Okay, so Darwin's theory is based on gradual process, small variations which are directional in nature and continuous in nature. Directional and continuous. That means chinna happening and continuously happening, not in a jerky manner, but in a continuous manner, okay? That is giving rise to new species. But Hugo de Vere says that no, it is by a single step, large scale mutation happened that has really changed the species, okay? Examples of natural selection. So you know that these are the examples of natural selection. Actually, natural selection can be studied as three types. Selection can be of three types. And let me take out that diagram directly so that I may not be drawing it on board. These are the three types of natural selection, as you can see here. Right? One is called the stabilizing one is called the directional one, and one is called disruptive. Okay? Stabilizing, directional, and disruptive. Three types. Examples of the stabilizing one, the birth weight of the babies. Now you will find that the mean value has not changed. Then directional one simply means that the phenotype will change in one direction. So one of the extreme becomes the mean value now. One of the extremes becomes the mean value, but only one will form. Okay, one species is formed. So this is called directional. But if both the extremes are increasing, okay, both the extremes, their frequencies are increasing, then we can have two mean values, and two mean values means two peaks. Two peaks means two different population, and these two different population eventually become two different species, and two different species out of one species. What it is giving you an idea. Something did you study just now? Something happened with the marsupials. You are noticing one thing happening here. Something happened to the marsupial. Something happened to the placentals in case of Australia. Something happened to the finches in case of Galapagos Islands. What is that? Adaptive radiation. Very good. So this is showing the radiation. ये क्या कर रहा है? देखो ये सेंट्रीफ्यूगल रेडिएशन, एक पॉपुलेशन से दो पॉपुलेशन बना, and then these two ultimately form two subspecies, and one day they will form two different species. In the same process, many different species are formed, and this is what adaptive radiation is all about. Okay. Now these are the examples. Examples of directional. So many examples of directional natural selection. Examples of directional are so many. For example, we can have now, if you understand this concept, you can identify in the given questions also. Now, we have tried all the type of question. I think they will not, we have not left any example, because this is a highly important area where questions are being asked usually every time. Okay, so they give some example and then they ask what type of evolution do you notice here. For example, they have given the birth weight of babies. Whenever the birth weight of babies are given, it is an example of a stabilizing selection. Okay, so whenever the mean value is advantageous or selected, it is always giving a stabilizing selection. That means the mean value has become stabilized. Okay, so other example of stabilizing one. So like for example, 2-3 tests before, we have given one question about the beak size. A very big beak size is not good. A very small beak size is also not good. So the type of food can be eaten by the moderate beak size. And therefore, you will find that the moderate beak size keep on getting selected. Again, stabilizing selection. Then directional, many examples. Our book has given some example. For example, any type of resistance, whether it is drug resistance or it is maybe any antibiotic resistance, antifungal, antiprotozoal, any drug resistance, whether any type of resistance against the herbicides. Herbicides are the agents used to kill the weeds, unwanted plants, weeds. We use herbicide chemicals, but now we find the development of resistance against the herbicide also. Any type of resistance, drug resistance, DDT resistance in the mosquitoes, again resistance, All type of resistance are examples of directional natural selection. Okay, that means one of the extreme value, which is a resistant quality, that has become the mean value now. All have become like that because all the sensitive one die. Only the one who are left are the non-sensitive or the resistant one. Then other example, for example, in 1 year they have given a question that we are selecting the cow on the basis of the yield of the milk. So whenever we are selecting the cow on the basis of the quantity of yield, what type of selection it is? Again, whenever we are selecting the cows on the basis of the quantity of the milk yield, we are calling it directional selection, because every time you are selecting only those cows who are superior. That means they give more amount of milk. Okay, they have higher yield, they give more amount of milk. Superior cow. So whenever you are selecting, you are always selecting the one with more, and only they are allowed to reproduce. So eventually the coming generations will have more of that type only, and one day that has become the mean value. And whenever we keep on selecting that, we find that the mean value is shifting in one direction, and that is called directional selection. Even industrial melanism is an example of directional natural selection. All adaptive radiations are example of disruptive natural selection. They can give examples, right? Okay, they can give example. For example, the beak of the blue-bellied nutcracker. Now, what has happened in the blue-bellied nutcracker? Earlier, the type of food that has now become scanty. So, what is available is either a very big seed or a very small seed. But their own food, that seed is now become scanty. So now only the survivors are having either a very bigger and strong beak or having a very pointed beak. So you will have two population, one population of a very strong beak and one population of very pointed beak. Because by a pointed beak, you can really pick up small seeds. And by a thick, big or bigger one, that beak you can really crush a bigger nut. So this is an example of disruptive natural selection, disruptive or centrifugal selection, and stabilizing is centripetal selection. I hope it is clear, everyone. This point is clear. Hardy-Weinberg is being disturbed by, can you recall that? What we have given that example? Mr. Michael does not revise. Mr. Michael does not revise. Now this trick we are using for the Hardy-Weinberg equilibrium. Now how the equilibrium can be disturbed? The equilibrium can be disturbed by 5 factors. What are those 5 factors? The 5 factors are whether there can be any mutation, whether there can be any migration, whether there can be any drift or genetic drift, whether there are natural selection or recombination. Okay, Mr. Michael does not revise. Mutation, migration, drift, natural selection, and recombination. Now, these are the 5 factors which can disturb the Hardy-Weinberg equilibrium. It means that because of these factors, the P plus Q is not equal to 1. But if the Hardy-Weinberg equilibrium is maintained, that means that there is no change happening in the gene frequency. Okay, all the genes of the gene pool, the frequency has remained same. Whether dominant allele or recessive allele, frequency is same. And frequency can remain same in a very, it's impossible, in fact. In a rare or of the rare also, but it is impossible, I can say, because you cannot stop mutation, migration, drift. These are the phenomena which usually happens by themselves. Mutation, you cannot stop, you cannot bring it. Of course, you can induce, you can try to induce, but you cannot stop them. Then migration. Now, whenever people migration will happen, and that is happening multiple times, this can give rise to a gene migration, okay? Then drift. Now this drift is interesting. The drift is it's an random change in the gene frequency. Whenever the change happens, that means the frequency changes of those genes which are giving them more fitness. That is called natural selection. But when those genes are increasing by itself, by chance, there is no adaptability or no advantage of those. They can be advantageous, they can be disadvantageous. Those are selected. That is known as drift, random change in gene frequency. Okay, so two type of gene frequency changes. One type in natural selection, only those genes frequency increase, which give them more fitness, which give them ability to utilize the resource better. But in case of drift, those genes are increasing in frequency randomly. Now this drift operates in a smaller population. It operates in a smaller population. Okay, the drift operates in a smaller population. A smaller population, because what is not happening in a smaller population is random mating. Because in a smaller population there is a assortative mating takes place, assortative mating or selfing, but not random mating. Random mating usually happens in a large population. Random mating. Okay, so either we can say that a small population not mating randomly, absence of random mating, small population, absence of random mating usually give rise to drift or genetic drift. Okay, bachcho? So they have added one more factor, which is small in size, bigger in size. So for change in the frequency or disturbance in the Hardy-Weinberg equilibrium, in which it is more chances of changes, smaller or bigger? Answer is smaller. Now, in which one you will have disturbance in the gene or in the Hardy-Weinberg equilibrium? Who will disturb the Hardy-Weinberg equilibrium? Random mating or assortative mating? Assortative matlab selective mating. Random mating or selective mating? Answer is selective mating. Random mating is not affecting the equilibrium, but selective mating is disturbing the equilibrium. Okay, so these 5 points, 2 more points are actually the outcome of this drift only, small size and also selectively mating, because random mating will maintain the frequency disturbance are these factors. So very important. Generally, I have seen that many people have remembered this very nicely. Very nicely. So when did you find the Dryopithecus, Ramapithecus 15 million years ago? Australopithecines are 2 million years ago. And Homo habilis, this is, the same time, roughly the same time, you can find this Homo habilis, that is 3 to 4 million years ago. Okay? No, Homo habilis is regarding 2 million years ago, 2 million. Homo habilis. Homo habilis has come out from Australopithecines. Australopithecine is 2 million years ago. Homo habilis is also of the same time. That is 2 million years ago. Okay? Then from that comes out the Homo erectus, that is 1.5 million years ago. Then Neanderthal man, that is about 1 lakh to 40,000 million years ago. And then the modern Homo sapien or the modern man, like anatomically modern Modern is not in terms of the mindset, modern in terms of the anatomy. Anatomically modern human that has evolved around 75,000 to 10,000 million years ago. Neanderthal man, 1 lakh to 40,000, and 75,000 to 10,000 is the modern human, Homo sapien sapien. Okay, please understand that this is also Neanderthal man is also Homo sapien. Okay, it is called Homo sapien neanderthalensis. So the cranial capacity is same. It is same as a modern Homo sapien. Same. 1400 cc. 900 cc is of the Homo erectus, like the Java man, Peking man. These are examples of Homo erectus. 1.5 million years ago, 900 cc. Fossil discovered in Java in the year 1891. Probably this is the first meat eater because it used the use of fire, and use of fire enabled him to cook the meat, and cooked meat is what these people can eat. Up to Homo habilis, Dryopithecus to Homo habilis. Yes, they fight, they can also kill each other, but they don't eat. They are not the meat eater. Most of them are frugivorous. They are fruit eaters. Okay? Neanderthal man, also like omnivorous. Okay? Modern Homo sapien is also omnivorous. But up to Homo habilis is all herbivorous. First time meat eater is Homo erectus. Neanderthal man lived near East and Central Asia and used hides to protect their body. First time someone is covering their body, and also the first time they are also burying their dead bodies. First time, religion, okay? The concept of rebirth, the soul does not die. Okay, Amaratma, that concept is first time developed. Today it is there in humans. But then, first time, Neanderthal man, that is why he is doing the ritual, the burial ritual. First time the burial ritual can be seen. Burial ritual, they use the ritual. They cover the dead body by flowers. Today pollens can be found over those graves, over the skeletons. We find the fossils of the pollen, fossilized pollens. So that ritual has been followed in all these dead bodies. That means burial ritual. It is the first time Neanderthal men. And this 18,000 years ago, prehistoric cave arts. 10,000 years ago, agriculture. Agriculture came about 10,000 years ago. But believe me, if questions come directly from NCERT, you can do the questions, okay? Have faith in yourself. You can remember that. But then if questions come on the basis, so those who have really done it earlier with many questions, then their chances are better. But if direct questions are there, I can say that more than 50% out there, they can recall these points. Right now, it is you are thinking, "Arey yaar, ye to bhul gaya tha. I have forgotten. Oh shit, I forgot Neanderthal man. I forgot this that." But then, right now, you are trying to recall so many things in a single point of time. But now, when single thing to be thought in the single point of time, that means that time the recall is easier. So in the examination, recall is easier. That is also there. Okay. Right now, recall, there's a false understanding that you are forgetting the things, but actually it is there in your subconscious mind, and you can, as a reflect, and you find this answer. You can find this. Many a times we change our answer, and the changed answer is wrong. If I have changed 10 times, 9 times, I make it wrong. Roughly the same ratio is in your case also, because first time whenever the answer come, it comes from the subconscious mind. And subconscious mind is what has developed by longer revisions and all. Okay, therefore that answer is usually the correct answer. But when you overthink, then you change it and then you make it incorrect. Okay, so please bubble also nicely. People are losing marks due to bubbling error now. They are saying, sir, देखिए ना, I have done no problem, sir. Only bubbling error. Bubbling error is not a no problem. It is a big problem. Beta, test is not the test of knowledge only. The test is of this ability also. How vigilant you are in bubbling. You know all the steps of the surgery. You know the steps. Someone is asking you, you are answering. But sir, sorry, that time I cut a different nerve. I couldn't see it is an artery or a nerve, sir, that time by chance. Okay, but then, sir, I know which artery to be blocked. Beta, but this is a question of life and death. Okay, so a doctor is supposed to be taking care of not only the knowledge, but these things also. So bubbling error is also equivalent error as an error in misunderstanding or silly mistake. Now, silly mistake is something which is, we can call them silly, but I don't know whether how silly they are. Silly matlab where something can be really ignored, but you cannot ignore the silly mistakes. The difference in marks of the first 25 people, the marks were same, no difference. Okay, same total 25 members. 715, 715. Right? If one silly mistake, you could have out of that top 25 category. One silly mistake. Two silly mistake, you are out of, because earlier the clustering is less, later on clustering is more. Similarly, as you go further at 620, 600, 590, 580, clustering matlab thousands of people with the same total. If any one of them does one extra mistake, he will be how many ranks behind? Thousands. Okay? One question means 5 marks. At each marks, we have many people. Every marks count. So, mistake is mistake. Don't categorize them as silly mistake or bubbling mistake. Okay? Because you have to learn all of them. You have to be careful in all of them. So senior, class 12th, we have got what is left is ecology. In class 12th, ecology is left. And class 11th, we have not done animal kingdom, structural or physiology. Tell me, I think people are answering more correct answers in physiology, unless the question is really complicated, but you are answering them well. What do you think so? Physiology में तुम लोग गलती कम करते हो. Either you have done it nicely or you understand the course easily. So intention errors are lesser here. Which one to do now? Which one? Chemical coordination. हाँ ये question पूछते हैं. Chemical coordination. Sir, we have forgotten the functions of the hormone. ठीक है बेटा. My health is good. My health is good. Chemical coordination done? Good. Sense organs you can ignore now. Sense organs you can ignore. So these are the, the conventional or the coordinated glands. Okay, so these glands, which are the endocrine glands, categorization of the endocrine glands was given in the very beginning as regular, categorization is not here, but I can tell you from myself. So we have got the organized endocrine gland, organized or the coordinated one. Organized these are the general glands that we understand, like pituitary, pineal, gonads, all the pancreas, all these are organized endocrine glands. But then some cells are there which are producing hormone, but they are not present in some endocrine gland, for example, like the atrial cells. Okay, they are producing the hormone, atrial natriuretic factor. Getting my point? Our GIT, stomach, duodenum, they are making many kidney, they are making hormone. These are called, unorganized endocrine glands, unregulated or unorganized endocrine glands. Okay, so this is the classification that you can see which of the following is an organized one and unorganized one. So generally by seeing the option you can identify. Don't goofed up ki sir they are asking about the organized. That means I thought they are asking about the paracrine or endocrine or heterocrine. No, not like that. Just whatever NCERT has given that classification. Now coming to the pituitary also called hypophysis. Okay, but above the pituitary in the brain you have got the hypothalamus, and above the hypothalamus in the brain you have got the pineal. Pineal gland is very easy to do because it is producing Pineal gland is producing only one hormone, and later on in the later stages of life, even this gland is also become degenerated and turn into some, some calcified tissue. Pineal gland, the hormone is melatonin, and it plays a very important role in regulation of 24 hours rhythm or the diurnal rhythm. Okay, maintaining the normal. Now, what are these diurnal rhythm? Sleep-wake cycle, sleep time, waking time, that is regulation of body temperature, changes in the body temperature with time. Okay, so that means there is some clock in the body. Where is that clock in the body which is actually taking care of this time? The clock in the body is not in the pineal gland, bachon. The master clock of the body is the hypothalamus. So hypothalamus is the master clock of the body. Hypothalamus. Hypothalamus is the master clock. Master clock of the body. Okay, hypothalamus. Pineal gland is only regulating the day night, that is only the sleep wake cycle and body temperature, but there are many other type of the rhythms. Okay, for example, something taken care in 7 days, 14 days, and something happened for 3 to 5 days, something happens only for 29 days. What happens only for 28, 29 days? What happens which is repeated every 29th day or later 28th day? I should say 29th day repeated. Yes, menstrual cycle. So even that is also there. Okay, so there are a lot of examples of the biorhythms. This is producing the hormone. Okay, but then in the brain we have hypothalamus, which is the master clock of the body. Regulation of body temperature, and it can influence other rhythms also. It can influence the rhythm in metabolism, because the moment we get up in the morning, the metabolism is low. It can change the pigmentation. Yes, pigmentation of in animals, in human being it is not very evident, but pigmentation is in other animal, you can see, controlled by the pineal gland. Pigmentation, menstrual cycle, as well as defense capability matlab immunity, the immune response. Okay, it can influence it regulate others and influence other. Influence you can forget, but don't forget what is doing the main function: regulation of body temperature, maintaining the normal rhythms of sleep wake cycle, and influence pigmentation, menstrual cycle, and defense capability. Okay, bachcho? Then we have got other in the brain, hypothalamus. After the pineal, we have the hypothalamus. Hypothalamus has got endocrine part also, and endocrine part of hypothalamus produce two type of hormones which are controlling the functions of the anterior pituitary hormone, and two hormones which are actually of the posterior pituitary, but they are manufactured inside the hypothalamus. They are released out by the posterior pituitary, but they are manufactured by the hypothalamus. Two of them. And the two type which are controlling the functions of the anterior pituitary, we are calling them, one type as releasing hormone and one type as inhibiting hormone. One example of releasing hormone is gonadotropin releasing hormone. But this is only one example. We have many such releasing hormones in the body which are for many other hormones of the anterior pituitary. Similarly, inhibiting hormone. One example of inhibiting hormone is growth hormone inhibiting hormone. And another name of growth hormone inhibiting hormone is somatostatin. Okay. So hypothalamus is easy because these categories you can remember, but one example of both the category you can remember. Okay, that two other manufactured in the hypothalamus will be actually secreted or released out by the posterior pituitary. These two hormones are ADH and the other one is oxytocin. ADH and oxytocin. Another name of ADH is vasopressin and oxytocin. These two are produced by the hypothalamus, but then they go to the posterior pituitary. Now, how these hormones are going to the pituitary and releasing out from the pituitary or controlling the pituitary hormones, okay? So, they can go by two ways, okay? Either they can go through the bloodstream, but the bloodstream, the normal course is a very long course. It can go into the main venous system, then vein into heart, then they will take a pulmonary circulation and then coming into systemic circulation but then this is all very long. There is a short circuit mechanism, and a short circuit mechanism is this one. This is a portal vein, the vein that begin from one organ and end into another organ, and they take blood from one organ and supply it to other organ. So this is called the hypophyseal portal vein, and it is taking, whenever we are using the name to the portal vein, we are actually naming the place where it is taking the hormone to. For example, if I am saying hypophyseal portal vein, that means it is taking the hormone to the blood is being taken to the hypophysis. The blood is going to the hypophysis. The vein is ending into hypophysis. That means pituitary, hypophyseal portal vein. So it is coming from the hypothalamus and going to the pituitary. Similarly, if I say hepatic portal vein, that means it is going to the liver. Renal portal vein, it is going to the kidneys. Okay, so hypophyseal portal vein, capillaries, and ending into here. The two type of hormone, releasing hormone and inhibiting hormone, and the two hormones which are ADH and oxytocin, they are coming through these neurons only, neuronally, and they can come to the posterior pituitary. So the two types are through the bloodstream or through the axons. Axons, okay. If they are coming to the anterior pituitary, they come through the blood, and if they are coming to the posterior pituitary, they come to through the axon. Okay? These are the hypothalamus. Releasing inhibiting hormones are produced by hypothalamus. GnRH, gonadotropin releasing hormone by hypothalamus. But who produces gonadotropin? And the answer is pituitary. The pituitary produces the gonadotropin. So next gland to do is the pituitary gland. The pituitary can be divided into anterior pituitary, middle pituitary, and posterior pituitary. Anterior and middle pituitary, they can be called as the adenohypophysis, and posterior pituitary, it can be called as the pars nervosa or the neurohypophysis. Neuro means nervous and or neuronal, and hypophysis means the pituitary. Adeno means glandular, hypophysis means pituitary. That means glandular pituitary or the nervous. Why it is called nervous pituitary? Because it is actually the neural extension of hypothalamus. It is just the neural extension of hypothalamus. Make sense? Now, can you recall all the hormone of anterior pituitary? All the hormones of anterior pituitary, what is that? Okay, fat pig, FATPIG, fat pig. F stands for FSH, then A stands for ACTH, adenocorticotropic hormone. Okay, after that we have got TSH, thyroid stimulating hormone. Okay, after that we have got P, prolactin. I is interstitial cell stimulating hormone, also known as, it is also called LH, luteinizing hormone. In male, we better call it as ICSH, but generally we are calling it LH. LH and ICSH are same. And afterward is PIG-G. G stands for growth hormone. Growth hormone can be called as somatotropin. Somatotropin. Somatostatin is growth hormone inhibiting hormone. Stato, ruk jao, growth hormone inhibiting hormone. And somatotropin is the growth hormone itself. somatotropin. And what are gonadotropin? The hormone that stimulates the gonads. So I hope you understand the word tropic hormones, the hormone that stimulate other glands. The gonadotropins are LH and FSH. LH and FSH are examples of gonadotropins. All pituitary hormones are actually peptides. Melatonin is amino acid derivative. Then hypothalamic hormone, all peptides, and here also all peptides. All pituitary hormones are peptides, all hypothalamic hormones are peptides, but melatonin is actually tryptophan, it is derived from tryptophan. So tryptophan derivative, but then other two glands, they are generally making peptide hormones in general. Okay, so then comes the middle lobe. Middle lobe produce the MSH, it is melanotropin or melanocyte stimulating hormone. Melanotropin, देखो tropin word हो गया ना, tropin means stimulating or melanocyte stimulating hormone. In human, it is a vestigial hormone because it is of no use. It is actually not doing anything in the humans, right? Then we have got posterior pituitary. The two hormones that come out from the posterior pituitary are ADH and oxytocin. But where they are manufactured in the hypothalamus. Where they are released out, from the posterior pituitary. And how do they come to the pituitary? They come axonally. Right? And so it is rightly said that posterior pituitary is just the neural extension of hypothalamus. So there are three main glands, and nothing is confusing in that: pineal, hypothalamus, and pituitary. These are main glands. Of course, these two, hypothalamus and pituitary, these are the main glands. Pituitary is the master endocrine gland, and hypothalamus is the master of the master. Right? So you can understand that it is father ka bhi father, master ka bhi master, hypermaster. What is hyperparasite? Hyperparasite क्या होता है? Hyperparasite पानी का है. What is hyperparasite? Parasites on the body of parasites. Very good. Parasites on the body of parasites are hyperparasites. Okay, I do take that. बच्चों, now coming to the other gland, and the next gland, in fact, these are the two glands at the same place. And I will ask you, where are these glands? First of all, I have to come in the camera to show you where I'm asking. I'm asking somewhere here. The two main glands here, thyroid and parathyroid, important glands. Thyroid, very, very important. Parathyroid, more important. Of course, for life, it is more important. So thyroid and parathyroid at this. And how many parathyroid we have? We have 4 parathyroid. How many thyroid we have? 1. But it is having 2 lobes. 2 lobes, and both the lobes are connected by isthmus. 2 lobes. Hypo, ये हो गया thyroid. Come. And thyroid is very easy to understand because what we forget are the allied functions. We don't forget the main function. Thyroid composed of 2 lobes located on either side of the trachea, the 2 lobes. Both the lobes are interconnected by a thin flap of connective tissue called isthmus. Okay. Then thyroid gland composed of stromal tissue made up of stroma cell. Stroma, that is a ground tissue. Other than that, we have the follicle, thyroid follicles. Okay. And follicles, the follicular cells are actually producing the thyroid hormones, the T3 and T4. Thyroid hormones, T3 and T4. Okay. Can I take a 2 minutes break? You can also take. You have been taking. Okay, so मार्कर को आ जाने दो. मार्कर Thank you for this break. Necessary. All good? Maybe you don't understand this. You may not understand this, but this has given me a lot of relaxation in life whenever I'm in anxiety. Still something is going on. It is dicey. It can go this side or that side. Like that. Okay, so I remember one poetic phrase. We are calling them shero shairi. So this word you might not be understanding, but some of you can understand. Those who know Urdu, they can understand this. क्यों सोचें जिंदगी में क्या होगा, क्यों सोचें जिंदगी में क्या होगा, कुछ ना होगा तो तजुर्बा होगा. Why keep on thinking what this money to this? At least some experience you will get. If something is mashed up, some experience it will give. तो क्यों सोचें जिंदगी में क्या होगा, कुछ नहीं होगा तो तजुर्बा होगा. तजुर्बा means experience. Some experience you will get. Okay, so हाँ जी बच्चों. Now anterior pituitary is also called anterior pituitary is also known as pars distalis. Middle one is pars intermedia and posterior one is pars nervosa. Pars distalis intermedia nervosa, DIN. Distalis intermedia nervosa. Distalis and intermedia both combine to form adenohypophysis, and nervosa is the neurohypophysis. Okay, done. Now we come to the thyroid gland. Thyroid use the thyroid hormone. Thyroid hormones are iodothyronines. They are iodinated thyronines, T3 and T4, triiodo and tetraiodothyronines. Thyroid hormones, what they are doing in the body, they are actually controlling the basal metabolic rate. The basal metabolic rate is the rate, which is required for our sustenance. With minimum metabolic rate that is required, basal matlab at the base. Throughout the day, the metabolic rate in the morning is different. In the middle of the day is different. Metabolic rate is fluctuating. After a strenuous exercise, metabolic rate is high. The basal metabolic rate is a bare minimum when we are at complete rest. So who is maintaining that? So who is maintaining our survival? Because without that metabolic rate, survival is not possible. So the basal metabolic rate is controlled by this hormone, triiodo or tetraiodothyronine. Both have the same effect. They are different only in terms of their strength, their potency. That triiodo is more powerful than tetraiodothyronine. T3 is more powerful than T4, and T4 is also called thyroxine. Thyroxine, so more powerful than thyroxine is T3 or triiodothyronine. Like that you have to understand. Other effect of the thyroid hormones, they are not there, never mind. I will tell you. Regulation of basal metabolic rate. Other effect is it also help in synthesis of RBCs, erythrocyte, erythropoiesis, right? Then number 3, the heat generation. It is also called calorigenic hormone, calorigenic, heat generating hormone, number 3. Number 4, it help in maintaining the ionic and water balance. Water and ionic balance maintained by the thyroid hormone. So these are the main effects of thyroid hormones. Okay, so thyroid hormones regulate, yeah, है ना ये और are here. Regulation of BMR, basal metabolic rate, erythropoiesis, RBC formation, metabolism. When we say basal metabolic rate, it is actually regulating the metabolism of carbs, proteins, and fats. Water and electrolyte balance, maintenance of that play important role in development of the central nervous system. Yes, so in people, suppose in the fetus, if the amount of thyroid hormone is less, right? If thyroid hormone is less, then it will result into retarded not only physical growth but also the mental growth. Yes, it also help in physical growth, but mainly it is helping in the development of the central nervous system. So if there is a scantier one, lesser one, then of course there is a problem, right? Then it can promote the DNA replication, that is cell division or growth. And one more function you can add from here is it help in transformation of the larva into adult, that is called metamorphosis. So now they can ask you, suppose if there is a deficiency of thyroid hormone in tadpole, what it will result into? Tadpole will remain as tadpole only. It will not become adult frog. Why? Because this hormone is essential. Now another way of giving the same question. Now they say that, okay, there is a deficiency of iodine. Same thing. Iodine deficiency also leads to the same problem. Deficiency of the iodinated hormones. उनका deficiency हो जाएगा, because iodine somehow we want out from outside, exogenous source. We don't produce this iodine from inside. It's a point clear to everyone. Please add this point also, that is metamorphosis, development of the central nervous system, mind. Therefore, there is a physical and mental retardation both in case of hypothyroidism. Better disorders of thyroid gland, both hyposecretion and hypersecretion. Hyposecretion, it can result due to deficiency of iodine in our diet. It will lead into the goiter, or it is also called endemic goiter. Endemic goiter because generally it is endemic to a region. It won't happen in one or two families. It will happen in the entire region. Endemic goiter, and this is because in the food chain of that place itself there is a deficiency of iodine. But today these endemic goiters do not take place. Why? Because everywhere, wherever they are using the salt, common salt for cooking or for mixing into salads, common salt is always coming with fortified with iodine. So the amount of iodine required for us is there in the common salt. So even if it doesn't come from any other source, common salt itself can fill up this gap. Cretinism, hypothyroidism during pregnancy, that leads into the physical and mental retardation both in the fetus. Okay. Now it will result into stunted growth, physical and mental growth both, low IQ, very very low IQ, subnormal IQ. Okay, abnormal skin and deaf mutism. What is deaf mutism? Congenital hearing loss, congenital deafness, that will lead to deaf mutism, congenital deafness. That will lead to deaf mutism. Deaf mutism. That means the person is not only deaf, but also there is no language. He is therefore mute, because what you learn is what you listen. What you learn is what you listen. Okay. Now it's just like, suppose if whatever I'm teaching, but from the very beginning the voice is not there. Really, even not only the classes, even the revision cannot go through, because hearing is so important. We have to hear what the other person is saying. So only by hearing we learn, right? Language particularly, the first word mama, nana, amma, these are we are learning when we are seeing people saying, and then we try to imitate them, we try to repeat them. So we hear and we learn. We hear, we learn. That is why even illiterate people, they can easily learn the language because they can hear. So they can learn that language because, okay, they don't know the script, but they can know the language, they can speak. this person is congenital deaf, by birth deaf, kabhi suna nahi anything. There is no language for him. Everything is. Also, don't say anything. This is called deaf mutism. It is a characteristic feature in case of cretinism. The word cretin, it means idiocy. Idiocy matlab, this is an idiot. Idiot means, idiot is not in derogatory word. Idiot is means subnormal IQ. Idiot is actually that category of the people having IQ from this range to this range. It is called idiot, right? That is subnormal IQ. Subnormal IQ, that is called idiot. Okay, bachcho. Cretinism, cases of hypothyroidism. Hyperthyroidism, that can occur due to nodule in the thyroid gland, cancer of the thyroid gland, okay? Due to nodule in the thyroid gland or cancer of the thyroid gland, hyperthyroidism. In hyperthyroidism, the level of the thyroid hormones is very high. And one such example of this high thyroid is an autoimmune disease called exophthalmic goiter or the Graves' disease. It is an autoimmune disease where the autoantibodies behave like the thyroid stimulating hormone or TSH. So the amount of TSH is actually low in the body only. It's lower than others, but these antibodies are behaving like TSH. And these antibodies are produced in abnormal quantity, and therefore this is leading to the excessive stimulation of this gland, leading to goiter. Now this is called exophthalmic goiter. This goiter is characterized by exophthalmos, that is eyeball, protrude out because of development of edema behind the eyeball. So it looks like that his eyes are almost out, okay? This is enlargement of the thyroid gland, and therefore we use the word goiter. So goiter simply means that galgant, there is a swelling in the neck, that is called goiter. Protrusion of eyeball, very high metabolic rate, and weight loss. Remember that low metabolic rate leading to weight gain and high metabolic rate leading to weight loss, okay? Hypothyroidism in adult female may cause irregularities of the menstrual cycle, because one of the function of this is to regulate the menstrual cycle also. Menstrual cycle, one of the function. Hyposecretion in case of adult female, that is leading to menstrual cycle irregularities. Parathyroid gland, very easy. Parathyroid, there are 4 parathyroid glands on the rear side of the thyroid gland. On the dorsal side or the back side of the thyroid gland, there are 4 small glands called parathyroid glands. So parathyroid glands secrete a hormone called parathormone. It is a protein hormone. Parathormone is a protein hormone, and it is actually regulating the level of calcium in the blood. That means it is increasing the calcium in the blood. So whenever the calcium levels are low, the body secretes parathormone to maintain the calcium level. So again, the calcium can come up, right? Okay. Now, another hormone that controls the calcium level is called TCT. TCT is called thyrocalcitonin. It is a hormone from the thyroid gland, but it is not a thyroid hormone. Now, it's so unusual to hear that what the sir is saying. It is from a thyroid gland, but it's not a thyroid hormone. Thyroid hormone are iodothyronines, triiodo, tetraiodo. But a peptide hormone is also produced from the stroma. From the stroma, a peptide hormone is produced, and that peptide hormone is the TCT, thyrocalcitonin. And TCT role is to decrease the blood calcium, and PTH role is to increase the blood calcium. So PTH increase the blood calcium, hypercalcemic, and TCT decrease the blood calcium or hypocalcemic. Now, they are not causing hypocalcemia as such, and neither the PTH is causing hypercalcemia, but that is an effect. That means whenever the body has low, calcium, at that time, what will release, PTH or TCT? PTH, to increase it, to make it to the normal level. And similarly, when the calcium levels are higher than normal, at that time the body produce and release TCT into the blood to decrease the calcium. Now, where is this calcium going? Immediately the body can deposit this calcium in the bank. And what is the calcium bank in the body? The calcium bank in the body are the bones. The bones are the calcium bank in the body. Bones. The point clear to you? Bones. So TCT is taking this calcium and deposit in the bones. PTH, what it does? It brings this calcium from the bones. This is called bony dissolution. Yeah, bone resorption. Reabsorption from the bone is bone resorption. Decalcification of bone decalcification. Calcium come out. So what all these names, they refer to the calcium leaving the bones and coming and filling this calcium level in the blood. This is being done by PTH, but then it is not the only role. If again PTH has to maintain this, it has got two more places to act. One place is the renal tubules, the uriniferous tubule, the nephron. At that place it will act, and if the body is releasing out calcium, if we are wasting this calcium, the body is dying of calcium, and we are peeing out calcium. No, PTH says, bro, no, stop. Don't release calcium. It will inhibit the calcium excretion through the urine. And third place it will do, it will help the body to absorb calcium from the gut. Now, you know that this work is only of the, which vitamin? Vitamin D. Vitamin D is essential for the absorption of calcium from the gut. But what the PTH is doing here, now PTH will make more amount of active vitamin D. Vitamin D we all have. We have got somewhat sunlight exposure. Our skin can make vitamin D. The amount that is required, that is enough for us. The skin can make, but the skin will make the initial product of vitamin D. Okay? The initial product. This initial product is being later on converted into active form, and active form is called calcitriol. I am coming to a very important question. Calcitriol. Thank you, beta. Thank you so much. Parathyroid hormone will cause the conversion of this vitamin D into calcitriol, which is active form of vitamin D, called D3 triol. D3. Active D3 will help in, so it look like PTH is absorbing the calcium, but it is not the, the PTH is acting on the gut. PTH is making more vitamin D, active vitamin D. And where is this active vitamin D formed? Active vitamin D formed in the kidney. Yes, skin produces that, but conversion into D3 occurs in the kidney and under the effect of PTH. Tell me one question. A question came, I think, 4 or 5 years back in 2019. Remember, 2019, one question came: if a person is using artificial kidney, what will not happen in this person, right? Now, what will not be seen in this person, and if he is using the artificial kidney, that means the kidney is not functioning properly, right? Then one line is about the calcium level in the blood. This person will have low calcium or high calcium. Low calcium or high calcium. Now, the answer is low calcium, because the way the PTH can act through the vitamin D, okay, because vitamin D is formed into calcitriol, the active vitamin D in the kidney, and this person has defect in the kidneys are not functioning. That means calcitriol is not forming. That means he is actually showing the sign of vitamin D deficiency. That is low calcium in the blood. So these people have to be given active vitamin D supplements. All people who are actually relying on hemodialysis, renal dialysis, they are given the supplements of vitamin D because their bodies cannot make active vitamin D. No matter how much they go in the sun, the active form conversion will not occur. And PTH is a key role. So PTH can work in 3 ways. Number 1, can take calcium from the bone and give it to the blood. Number 2, can take from the urine, give it to the blood. And number 3, can take from the gut, from the food and give it to the blood, somehow increasing the blood calcium levels. TCT will take this calcium and give it to the bone, decreasing the blood calcium level. And what we want to be there in the body, we want equal balance. We want a balance of TCT and PTH. So calcium homeostasis is maintained jointly by PTH and TCT. Okay, bachon, is this point clear to everyone? Can I ask you one question? Name the gland that produce TCT or the calcitonin. Which gland produce TCT? Which gland produce TCT? क्या हुआ? चैट नहीं आ रहा? TCT, calcitonin. Sumajra, what happened, beta? Slept properly? Thyroid, yes, thyroid. Good, thyroid. Thyroid, okay, thyroid, yes. TCT, calcitonin. The name is thyrocalcitonin from the thyroid gland, and PTH, parathormone, from the parathyroid gland. Name the gland that produce thymosins. Thymosins. Lot of sins, not one sin, many sins. Thymosins. Name the gland. Name the gland. Anish, you deleted the answer, but your answer was correct. Thymus, yes, thymus produce thymosins. And this hormone is required for the development of or the maturation of the T lymphocytes. And also, it helps in the formation of antibodies by the B lymphocytes. So both. Of course, not differentiation of the B lymphocyte, but help in the antibody formation by the B lymphocyte. And number 1, differentiation of the T lymphocytes. So in a way, it is a very important hormone for acquired immunity. As we age, thymus is degenerated. It is maximum at the time of birth. After that, it starts degenerating. I think in my body, by the time my body, there is no thymus. I mean to say, it is maximum at the time of birth. And from that time, it is degenerating. So many years now. Okay, almost going to be half century up. Another 2 years, half century up. Okay, so yeah. Your body also, but much better than my body. Your body is still good. Now tell me, who will have better immunity, you or me? Obviously you. And as I age further, the immunity will go down more. Now, one of the theory of aging is saying that it is not that you are losing your immunity because you age. No, you age because you lose your immunity. The reason why we age is because of the Immunity, the immune system is decreasing, and this is the reason for aging. The immunity does not decrease because we age. No, we age because the immunity decrease, because this gland, it is actually decreasing in size as we are aging. So tell me, who were given the shots of the corona vaccine first? The old people. सबसे पहले young तो very they have got good immune system. Old people 50 plus, 60 plus, their immune system is really compromised. उनको पहले immunize किया. Okay, the government immunize them first. Then come the next bracket. Then comes the younger one, like your age. Okay, so my age was in middle. Earlier, my parental age, then my age, and then came your age. और जो अभी born हुए हैं, right? वो पता नहीं, they want, they can take. But that is not so much essential for them to take, because after all, they will develop that. They will develop this immunity. Okay, you understand that? So as aging is seen, we find that the immune system is also weaker. So why we age? This is one of the theories of aging. Okay, but this line is, I think I have read somewhere in the books only. Old individual thymus is degenerated, decreased thymosin production, that is weak immune system. So why the old people have weak immune system? This is the answer to that. But so now comes the another gland, is adrenal gland. This is important, adrenal gland, very, very important gland. Now there are two glands here, one is cortex and one is medulla. Adrenal medulla is extension of the sympathetic nervous system. Adrenal cortex is mesodermal gland. So these are actually two different glands. One is ectodermal, that is medulla, and one is mesodermal, that is cortex. So this is an example of a gland within a gland. Adrenal gland, gland within a gland. Let's start with the adrenal cortex, because if you don't have adrenal medulla, then you will not have any problem to survive. You can easily live your life. But if someone has no adrenal cortex, then he cannot survive for even one day without adrenal cortex. But without adrenal medulla, you can survive without adrenal medulla, because it is just an extension of the sympathetic nervous system. Adrenal cortex, bachcho, this is having three layers from outside to inside. So outer side is one zona, another zone, and then the third zone, three zones. And then comes the medulla. Cortex is called the 4S gland. It is called the 4S gland. This is salt, sugar, sex, stress. Salt, sugar, sex, stress. First of all, from outside to inside, we can say salt. Outer zona, that is for salt. Middle zona for sugar. Inner zona for sex. Salt, sugar, sex. Zona glomerulosa. Zona fasciculata. Zona reticularis. Glomerulosa fasciculata reticularis, GFR. Glomerulosa fasciculata reticularis. Glomerulosa produce the aldosterone. What is that called? It is called mineralocorticoid. Then fasciculata produce the glucocorticoid, and this one is producing the sex corticoids, or the adrenal androgens. That is a source of the androgen in female is the adrenal gland. Source of androgen in female is the adrenal gland. Yes, females also have testosterone, but poor males, they don't have progesterone and estrogen. Males में progesterone estrogen नहीं होता. Normal males में they don't have progesterone and estrogen, but females have androgens. And the source here is, of course, there are two sources, but then one is beyond your scope. The other one you can understand is the adrenal gland. We'll continue till 1:10 तक कर लो तो अच्छा है. 1:10, 1:15. I know you are hungry, but till 1 you can complete. You can continue. Okay, बच्चों. Mineralocorticoid. Example of mineralocorticoid is aldosterone. Example of glucocorticoid is cortisol and androgens. So the names are not important here. They are actually different type of testosterone, DHEA. Okay, so they are different type of testosterone only. Androgens. Okay bachcho. Now more important than this GFR is the outer zone for mineralocorticoid, middle zone for glucocorticoid, inner zone for sex corticoid. The glucocorticoid also important for several things. It can increase the blood sugar level. It is hyperglycemic hormone. It is also causing the RBC production. It takes care of RBC production. Okay, hyperglycemic hormone. Where it is written, we can do it from there. Number one. Number two, what else? The action of cortisol. Number three, cortisol is it is anti-allergic, anti-inflammatory, right? Now, please understand that how it causes hyperglycemia. Try to think how it causes. I'll tell you the answer, and that you have to remember. It causes hyperglycemia by gluconeogenesis, by one mechanism called gluconeogenesis. That means new glucose formation. Ante non-carbohydrate se carbohydrate banana means from fatty acid or amino acid to glucose, right? This is the gluconeogenesis. It requires fatty acid, raw material, fatty acid, amino acid. How to obtain the raw material? You can obtain the raw material by proteolysis and lipolysis. Digest these fats, take the fatty acid from them, digest the protein, break the protein chain and take the amino acid from them. Proteolysis, breakdown of protein, lipolysis, breakdown of lipids. So you will get but then one problem, there are some cheaters, they will steal your amino acid. And who are them? The body cells. The body cell, they will see who is moving in the blood. Amino acid is moving in the blood. Get that amino acid. What we'll do? We'll make something. It will be required to make some protein. Don't worry. We will do something out of it. Bring that amino acid. Cells can take up this amino acid. Cortisol has to bring it to the liver to convert it into glucose. But these cheaters are there in between. So cortisol is making some strategy. It is actually inhibiting the uptake of amino acid from the blood. So it decreases amino acid uptake, utilization of amino acid, uptake of amino acid. So I think there's a line written in the textbook, cellular uptake and utilization of amino acid. It decreases or inhibits cellular uptake and utilization of amino acids, right? Because it has to protect these amino acids from being stolen by the cells. These are the effects, okay? So number one, please remember hyperglycemia. And therefore it is called the stress hormone. Why it is called stress hormone? Because under stress, or what is stress to the body? One stress that we understand is a mental stress, okay? That is in English language we use the stress, but one stress is the medical language we use. Okay, so what is the body stress or the physiological stress. But so the physiological stress is when the need of the energy is increased in the blood, it is called physiological stress. And it is overcoming that need. It is increasing the blood glucose at that time. So therefore it is called the stress hormone. It does not call mental stress. It is a physical or physiological stress. The need for energy is very high. Okay. And yes, this is the reason, inherent reason why it helps in RBC production, so that oxygen can be brought to the cells nicely. So these are the effects of the cortisol, so many effects. Coming to the effects of the aldosterone, only one effect. It helps in sodium and water reabsorption from the distal parts of the nephron, distal part of the nephron, distal part of the tubule, salt, sodium and water reabsorption, aldosterone. But if you have to choose out of the two, one, what actually it is doing? Sodium reabsorption. Water reabsorption is, after all, it is passive. Oh, it is diffusion, it is passive, osmosis. Wherever sodium is increased, at that place water will also increase now. So if you want to increase water there, don't work on water. No, you cannot work directly on water. You increase sodium there, automatically water will be taken there. So when body wants to regain water, okay, or keep more water, it will increase the level of salt. As the salt increase, as sodium increase, water will also increase in the blood. Okay, this is a strategy. So mainly help in the sodium and water reabsorption, but directly helping in sodium reabsorption. But if we have an option like sodium and water, then of course that is a more NCERT relevant answer. Okay, bachcho, that is mineralocorticoids. Uh, Sex corticoids are only androgens, and they are helping, they are playing a role in the growth of the hair. Okay, so axillary hair, armpits, okay, and the pubic hair, axillary hairs. Axilla means armpits, so mainly the adrenal androgens. Okay, bachcho, so that's about the androgens, adrenal androgens, and that is about the, uh, you know, adrenal gland, adrenal medulla. Adrenal medulla produce catecholamines. The two main catecholamines which are produced by the adrenal medulla, norepinephrine and noradrenaline. Sorry, norepinephrine and epinephrine. Norepinephrine and epinephrine. Epinephrine, you can use the word adrenaline for that, because in market it is marketed as adrenaline. So adrenaline is a market name, brand name, injection name. Hormone name is epinephrine. So in books it is for same. Epinephrine is same as adrenaline, norepinephrine is same as noradrenaline. First of all, understand. Also understand that both of them have the same effect. The effect on the body tissue is in the same direction. They are not in opposite direction. They are doing the same. They are brothers, right? So they are having the same effect on the body. And what type of effect they do? They are causing the effect of the sympathetic nervous system. That means they can increase the alertness. Yes, they can also increase. They can also increase because, sympathetic nervous system is for fight and flight response. That means it can help the body to overcome with the emergency. It is also called emergency hormone. Catecholamines, emergency hormone, fight or flight hormone. So in emergency, we find there is a more need of energy. Okay, and at that time, it is not a time to make. It is a time to use from the our stored sources, and the stored form of glucose is the glycogen. Okay, so basically this help in the breakdown of glycogen, and that's how the glucose can be taken in the blood, right? Breakdown of glycogen. So what that can be called? That can be called as glycogenolysis or glycogen lysis, and between there is a O word. Glycogenolysis. That become glycogenolysis. Glycogenolysis. Glycogen breakdown. Okay, because when I have enough of glucose, we can store it for the time of need. Now the time of need has come. Now bring that, and then at the time of need, the body produce epinephrine, norepinephrine, that will help in glycogenolysis. Where is this glycogen is stored in our body? Where we have glycogen? Where? In which part glycogen is there? Brain, liver, kidney, heart, skeletal muscles. Skeletal muscles, liver, kidney, brain and heart. Where you will find? The two answers are there. Tell me two answers. One is liver. Okay, I take that, liver. So one place where glycogen is stored in the body is the liver. Do you know any other place where glycogen can be stored in the body? Any other place where glycogen is stored? One is liver. The other answer is muscles. Yes, you are right, muscles. In the skeletal muscles, glycogen can be stored, muscles. Okay, so muscles and liver, these are the two forms, two places where the glycogen is stored. So it helps in increasing the blood sugar or hyperglycemia. And how? What is the cause of hyperglycemia now? Glycogenolysis. Cortisol also cause hyperglycemia. But then by what mechanism cortisol will do hyperglycemia? Cortisol will do hyperglycemia by gluconeogenesis. Okay. So now tell me the name of the hyperglycemic hormone. Hyperglycemic hormones. Hyperglycemic hormones. Tell me, number one, hyperglycemic hormone. One that we already know is glycogen. Opposite of insulin, glycogen is there, hyperglycemic. Any other hyperglycemic hormone? Just now we have done cortisol. Very good. One is cortisol. Any other that we know? Any other? Just now we have done, that is catecholamines. Catecholamines are epinephrine and norepinephrine, adrenaline and noradrenaline. Hyperglycemic, increase the blood sugar level. Any other that can increase the blood sugar level? Any other hormone that you know can increase the blood sugar level? Glucagon already we have done, cortisol we have written, catecholamines, epinephrine, norepinephrine already we have written. Any other? ठीक है, okay. Then let me bring to you the hormone that will increase the erythropoiesis. Erythropoietic hormone. Erythropoietic hormones. Can you remember the erythropoietic hormones that will cause erythropoiesis? Yes. Number one is thyroid hormones. T3 and T4. Thyroid hormones. Yes, it will cause erythropoiesis. Cortisol will help in erythropoiesis. Cortisol. Any other hormone? A hormone from the kidneys that you know? Hormone from the kidneys. That is, yes, you're right. That is, yes, erythropoietin. Any other? One more. Testosterone. In the summary, you can find testosterone. Thyroid hormones, cortisol, erythropoietin, testosterone. These are erythropoietic hormone. Erythropoietic. Now tell me, hypoglycemic Hypoglycemic hormone. Hypoglycemic hormone. Very, very easy. Utterly easy. Hypoglycemic. The another one is hypocalcemic. Hypocalcemic. Okay. Hypoglycemic, yes. Yes, you're right. This is insulin. Insulin. How many chains are there in pro-insulin? 3 chains, A, B, and C. And mature insulin, 2 chains, A and B. How many amino acids are in the chain 1? The first chain. How many amino acids are there? Chain A. How many? 21. How many are in chain B? 30. Hypocalcemic, hypercalcemic, PTH. Hypoglycemic, insulin. Hyperglycemic, 3 names. We have suggested you 3 names. Erythropoietic, 4 names. We have suggested you 4 names. Okay. Tell me which hormone can increase blood sodium level, which hormone can decrease blood potassium level, decrease the blood phosphate level. Which hormone can decrease the blood phosphate level? So on one side is hypernatremia, hypokalemia, hypophosphatemia. Phosphates come, then potassium come, taqwa, and sodium, aqua. All 3 one hormone only, yes. All 3 one hormone. And that one hormone is, yes, aldosterone. Aldosterone. So aldosterone, all 3 are doing, all 3 are the functions of the one hormone, aldosterone. Increase blood sodium, decrease blood potassium and blood phosphates. Makes sense? Aldosterone. Reabsorption of sodium, elimination of potassium and phosphates. ठीक है बेटा. So this is one thing. Then after that, because endocrine chapter 2-3 of you have spoken. Endocrine, हाँ मतलब ठीक है. It looks like that, because so many examples, so many effects are given. So always it look like that we are forgetting some of them. But you will notice that majority of the questions will be asked on the major effects only, the major effects. Okay. Coming to the adrenal gland. Catecholamines. Another important gland what is left is this one, that is pancreas. I think that is another gland that we usually forget the effects of insulin. Effects of insulin. Glucagon is easy, but effects of insulin are also easy. Okay. Let me tell you the effects of insulin and glucagon. Hello. Pro. Come on. क्या हो गए? As if the board is saying, enough, yaar, enough. No board, this is marathon. Hello? बोर्ड बोल रहा हो गया भाई, नहीं, मैराथन. This is what happens if you keep on, storing your urine. It will abort by itself. This is the, after some time, the voluntary control is gone. Although maturation is voluntary, so do defecation. But after a certain time, the voluntary thing becomes irrelevant. It come into the auto mode. ठीक है. Any other chapter of human physiology? Pancreas, I can do it without this only. Insulin. Now, insulin is hypoglycemic hormone. What it is doing, it is actually decreasing your blood glucose level. Now, how it can decrease the blood, why it is decreasing the blood glucose after all? See, actually, this is the after effect of its main effect. The main effect is it has to make your cells absorb the glucose. Right. Glucose cannot enter our body cell by themselves. There are no special channels by which glucose can enter by themselves. Human health, promise, I'm coming. There is no special channel by which, last thing, open कर लेंगे. Human health, I will complete. ठीक है. Ecology. Which chapter? Okay. Okay. So, glucose, the entry of glucose is actually made with the help of insulin. Insulin will translocate certain proteins to the membrane, and this will make the membrane permeable to glucose. And that's how the glucose can enter into the cell and the body cell can utilize it. So insulin is very important for the cell to utilize this glucose. If there is no insulin, the body cell can never take up. So it helps in the cellular uptake of glucose and of course also utilization of glucose, both uptake and utilization. Uptake and utilization. Without insulin, uptake is even not there. So if the cells cannot take up from the blood, it keep on building up in the blood. It keep on building up, cause hyperglycemia. Clear? So I think this point is clear. So this is the main effect. Then insulin is actually promoting conversion of this glucose into glycogen. Okay, that is glycogenesis. And glycogen is stored in the liver and the muscles. Glycogen. Insulin help in the conversion of glucose into glycogen. And insulin receptors are there in your liver cells and in your adipose cells. Liver cells and adipose cells, fat cells. Insulin receptors are seen. If the receptors become faulty, they don't respond. That is called insulin intolerance. Okay, you understand this point? Okay, so if the insulin deficiency occurs due to some autoimmune damage to the beta cell, by birth beta cells are lesser, insulin deficiency is there, then this is an example of by birth diabetes mellitus. Some people will develop this diabetes mellitus after some age. These are the people who have got autoimmune damage to the beta cell. So they will develop after a certain age, right? These people will respond to treatment of insulin. Their diabetes mellitus is called insulin dependent diabetes mellitus. We can also call it type 1 diabetes mellitus. Another type of diabetes mellitus is type 2 diabetes mellitus. And this is not due to any problem in the insulin. It is due to lifestyle. Due to lifestyle. Okay, insulin is enough, but the body cells, because of the poor lifestyle, the body cells they become resistant to insulin. They become resistant to insulin. Okay, so insulin resistance, we can say, or simple word, the body cell become resistant to insulin. Insulin comes, but it cannot work because the receptors by which the insulin will work, it will not be there. So prolonged hyperglycemia is seen in diabetes mellitus. One line of the NCRT has been corrected, I think, during editing. It came and the meaning coming out to be that if there is a prolonged hyperglycemia, it leads to it leads to diabetes mellitus. Hyperglycemia leads to diabetes mellitus. Now, this is a little faulty statement to make. Hyperglycemia is a result of diabetes mellitus. It's not the cause of diabetes mellitus. Hyperglycemia is it result due to diabetes mellitus. Whether the type 1 diabetes mellitus, which can be due to problems in the insulin, or type 2 diabetes mellitus, which is due to insulin resistance or type 2 lifestyle because of poor lifestyle. Then in these people, they will have the energy from some other source. So what happens? Their fatty acids are converted. The fatty acids are converted into the ketone bodies, and their bodies can take energy from the ketone bodies. So ketone bodies are providing the energy what the glucose cannot do. So glucose is all waste. The glucose go waste. In fact, it will cause hyperglycemia, and ketone bodies build up. Ketone bodies build up. Sometimes these ketone bodies are more than what is required at that time, and they will come because they can be simply filtered by the kidney and not reabsorbed back. They can be filtered. So that will come in the urine, that is called ketonuria. Ketonuria. If people have increased glucose in the blood, that will also come in the urine as glycosuria, glycosuria and ketonuria. Now, when you find ketonuria with glycosuria, it seems that the person is suffering from DM. Person is suffering from DM, you will find ketone bodies and glucose both. But if if you're only finding ketone bodies but not glucose at all, okay, in fact, you find that the blood glucose level is barely minimum. Even if you take the blood test, the blood glucose level is barely minimum, but ketone bodies are there in the blood in high amount. Now, this is a different case. In this case, why the ketone bodies? Of course, the body is obtaining energy from some other source, alternate source, right? But in this case, it is not because the body is unable to utilize glucose. No, it is because the body does not have enough glucose. These are the people of starvation. So if people are with starvation, they will develop starvation. Starvation means they are not eating the food, or they are fasting for a long period. They are prolonged fasting, starvation, prolonged fasting. Prolonged fasting is also leading to ketonuria, but they will have low blood glucose, ketonuria with hypoglycemia, okay. But if there is a ketonuria with glycosuria, glucose in the urine, ketonuria, ketones in the urine, glucose, glycosuria, glucose in the urine. Now this is a different case. Now first case, because this is due to starvation, and this is due to diabetes mellitus, ketonuria plus glycosuria, diabetes mellitus. But if the question is given that simply they will ask you ketonuria can be seen in number one only in case of diabetes mellitus, number two in case of starvation, number three in case of hypercalcemia, okay. Number four, 1 and 2, both 1 and 2. In this case, the best answer will be 1, 2 or 4. Which one? You know that third one is just like that only. No need, Chinmay, right now not required. Very few chances they can give some question on that. We have not seen frequently, like abhi to 8 to 10 years we have not seen questions coming from those cladograms. No doubt we have done in the class, in the revision also we have done, but at this time, those who remember that time, it's fine. But at this time, of course, correct answer is, the question is ketonuria. Only ketonuria is given. Ketonuria. So obviously, ketonuria can indicate both either diabetes mellitus, also due to starvation. So the correct answer is fourth. It can be due to both, because you can find that in both. Okay. But if the question comes ketonuria with glycosuria, then of course that is due to diabetes mellitus. So like that you should be able to answer the question. Simply ketonuria, we have an option which says starvation and DM. Obviously the best answer is that only, both starvation and DM, diabetes mellitus. Okay. Now, as you are saying, the human health and disease. Now, some important diseases, some common human diseases are bacterial one are typhoid, okay, typhoid or enteric fever. It is due to Salmonella typhi, and pneumonia. It is due to two bacteria, Streptococcus pneumoniae and Haemophilus influenzae. Streptococcus pneumoniae and Haemophilus influenzae. These are the two bacterial one. Okay, then other bacterial include dysentery, plague, diphtheria, tetanus, tuberculosis, whooping cough, and cholera. Dysentery due to E. coli, plague due to Yersinia pestis, diphtheria due to Corynebacterium diphtheriae, then tetanus due to Clostridium tetani, then tuberculosis due to Mycobacterium tuberculosis, whooping cough due to the whooping cough, that is pertussis. This is due to Bordetella pertussis. Where is the marker? This is due to Bordetella pertussis, whooping cough. Bordetella. If you have done it earlier, then it is good for you to just remember Bordetella. pertussis. Pertussis. Bordetella pertussis. Whooping cough. Cholera. Cholera due to cholera. Vibrio cholerae. Vibrio cholerae. These are the different examples of the bacterial disease. Common cold, chikungunya, dengue, okay, hepatitis B. These are some common viral disease. Common cold is due to rhinovirus, common cold. Chikungunya is due to chikungunya virus. Dengue is due to flaviarbovirus, okay, flaviarbovirus. Both these examples are the examples where the mosquito vector is transmitting this virus from a diseased person to the healthy person. The mosquito is Aedes aegypti, also called tiger mosquito. Aedes aegypti, also called tiger mosquito. Okay, dengue fever, also known as break bone fever. Okay, flaviarbovirus. Okay, then chikungunya. Of course, both are fevers. Severe frontal headache. मत करो इतना, ठीक है. Both are the common feature you will find is fever. Hepatitis B virus, HBV, hepatitis B virus, severe liver damage. Whenever liver damage occurs, jaundice result, recombinant DNA vaccine. By recombinant DNA technique in yeast, the vaccine is produced by the yeast. In the yeast, recombinant DNA vaccine in the yeast transmits through parenteral and sexual route. Parenteral matlab parenteral. Parenteral means other than enteron. Other than enteron means blood to blood, okay? And sexual route. Yes, hepatitis B is also an example of STD. And also it can cross the placenta. It can cross the placenta from mother to the fetus, just like the AIDS virus can do. Other viral disease like polio, influenza, measles, chickenpox, mumps, rabies, swine flu, COVID-19, SARS, severe acute respiratory syndrome, SARS. All these are examples of viral disease. So bacterial disease, viral disease, you have to identify which group it belongs to. Then protozoal disease. Some examples of protozoal disease. Number 1 is malaria. Okay. Number 2, protozoal disease, already we have seen this name, and the name is Trichomonas vaginalis, Trichomoniasis, it is an STD, sexually transmitted disease, protozoal disease. One is malaria, then number 1. Number 2 is trichomoniasis, trichomoniasis, number 2. And number 3 is can you tell me the answer? This is very common in AIDS patients. Other than that, people don't develop that. We are so immune that our immune system cannot allow this disease to happen. This can enter into the body. This protozoa, the name begins with T. T for typhoid, T. Anish Arya. T. एंटामीबा तो ई हो गया ना. T. Entamoeba, yes, but entamoeba occurs in any case, beta. Entamoeba is not only in the AIDS patient. In AIDS patient particularly, tuberculosis is bacterial. Beta. That is toxoplasmosis. Toxoplasma gondii, toxoplasmosis, toxoplasma gondii, and amoebiasis, entamoeba histolytica, trichomonas vaginalis, and toxoplasma gondii. So the name of the protozoa is very easy to remember. In fact, just remember the name of the protozoa. The name of the disease is easy to remember. So if you remember the protozoal name, amoebiasis. If you remember trichomoniasis. If you remember toxoplasmosis. So if you remember the name of the protozoa, you can easily recall the name of the disorder. Okay, these are the protozoal one, and fungal one is a ringworm. Ringworm is not a worm; it is a fungal disease. It is produced by or caused by or the pathogen three genera of the fungi, and these three genera are MET, not here we have MET, that is Mycosporum, Microsporum, Trichophyton, Epidermophyton, Microsporum, not Microsporum. कंफर्म करो ये माइक्रोस्पोरम है माइकोस्पोरम है ट्राइकोफाइटोन एपिडर्मोफाइटोन MET माइक्रोस्पोरम एपिडर्मोफाइटोन ट्राइकोफाइटोन ट्रिपेनोसोमा अनदर T yes sleeping sickness good माइक्रोस्पोरम ठीक है माइक्रो गुड माइक्रोस्पोरम ठीक है ओके तो this is not the time for that I think. Now let me discuss with you some drugs, and the drugs which are commonly abused are opioids, cannabinoids, coca alkaloids, and nicotine. Okay, nicotine, that is nicotiana, tobacco. The agent is nicotine. Drugs and alcohol abuse, right? So here we have opioids. Examples of opioids are morphine and heroin. Morphine is a painkiller, world biggest painkiller. Heroin, diacetyl morphine is heroin. Morphine is obtained from the latex of poppy plant, Papaver somniferum. From the latex we obtain morphine, and diacetylation of morphine is heroin. Now what it can do, morphine extracted from the latex of poppy plant, heroin commonly called as SMAC, diacetyl morphine, white, odorless, bitter crystalline compound, white, odorless, bitter crystalline compound taken by snorting and injection. Morphine is sedative and painkiller, useful in patients who have undergone a surgery. Heroin is a depressant and slow down body function. Heroin is a depressant and slow down body function. It is also called SMAC. And this tetracyclic diagram, and this opium poppy, the plant, poppy plant, okay. Then we come to, this is one category. इसमें one thing is left, which is the receptors. Opioid receptors are present in the CNS and in the GIT, both. So here we have CNS and GIT, both the receptors are present and taken by snorting and injection. Coming to cannabinoids obtained from cannabis sativa, bhang, charas, ganja, marijuana. These are cannabis, bhang, charas, ganja, marijuana. Okay, features. Flower tops, leaves, and the resin of cannabis plants are used in various combinations. So the female inflorescence, flowers, leaves, the resin, they are used in various combinations, and it can be taken by inhalation or by oral ingestion. Inhalation, oral ingestion, same thing can be smoked, and same thing can be added into the milk. Yeah, or curd. Interact with receptors present in the brain, and it shows effects on cardiovascular system of the body. This is the effect, but then it has got which category it belongs to? It belongs to hallucinogen. It is a hallucinogen. It will cause hallucination. It is a hallucinogen. Hallucinogen, it will cause hallucination. Hallucination. Okay. Hallucinogen. See the diagram, the chemical diagram, a central ring, C for center, C for a ring in the center, C for cannabinoids, and you can see the leaves, okay, the leaves of cannabis sativa. Coming to the coca alkaloids, example cocaine, cocaine taken from or obtained from the leaves of Erythroxylum coca plant. इसकी leaves है Erythroxylum coca leaves, commonly called as coke or crack, native of South America. Yes, it is principally by snorting. Cocaine को snorting से करते हैं. Inhalation, interferes with transport of dopamine. The neurotransmitter dopamine shows potent stimulating action on the CNS. Yes, it belongs to the category of stimulant, depressant, then hallucinogen, and it is a stimulant, stimulation of the CNS. Stimulant, it belongs to a stimulant category. Produces sense of euphoria and increased energy. Euphoria, happy feeling, excessive happiness, increased energy. Excessive dosage of cocaine can cause hallucination. So the normal dosage is taken for increased energy, but if you take in excess, then you will go into a different state of mind. Hallucination. Okay, done. Other drugs are datura, from the datura plant, datura stramonium. Datura is the name of the plant. Datura stramonium, and Atropa belladonna, deadly nightshade. These the products are, they are hallucinogens. Okay. Another hallucinogen is obtained from a fungus. Fungus. And this is actually a Mexican mushroom. It's a Mexican mushroom. And the name is, okay, I'll tell you the name of the compound. Then you can tell me the source. It is called LSD, lysergic acid diethylamide. Just remember LSD. LSD you can remember as life-saving drug. LSD, life-saving drug. It is not life-saving drug. It is lysergic acid diethylamide. Americans call it life-saving drugs, LSD. It is obtained from a mushroom. Any name? ठीक है, NCERT में नहीं दिया. Anyone know the name? अच्छा, if you remember this name claviceps, then it is okay. Otherwise, if you have never heard the name claviceps purpurea, never heard this name, don't try to learn right now. Even if you try, you will not remember it. But if you remember, if you have done it earlier, claviceps purpurea, this is the name of the Mexican mushroom. And you know that solution which I was thinking of in the DNA to for density gradient centrifugation, that solution is CaCl, cesium chloride solution, cesium chloride. Now the name is coming, cesium chloride. Okay. Tobacco. And some drugs are actually therapeutic drugs, but then therapeutic matlab they are used in therapy. They are used in treatment of depression, insomnia, treatment of depression, anxiety, insomnia, and these are barbiturates, benzodiazepines, amphetamines. Barbiturates, benzodiazepines, amphetamines. Okay, so please remember BABA, benzodiazepines, A for amphetamines, and BA, BABA, barbiturates, benzodiazepines, amphetamines, barbiturates, BABA. BABA has become bye-bye. Why? Because of their misuse, they are being banned all over. In our country, they are, I think everywhere. Everywhere it is banned. It is banned. Now it's a global world. So if anything is harmful here, it is actually harmful everywhere. So people will ban, all the governments will ban it. Baba become bye-bye. Barbiturates, benzodiazepines. Baba matlab B, benzodiazepine, A, amphetamines, and BA, Baba, that is barbiturates. Used as medicine to help patient cope up with mental illness like depression and insomnia. They are therapeutic drugs, but then misused. Tobacco, used by human being for more than 400 years. Smoked, chewed, or snuffed. I think these things you know already, right? Smoking, increased carbon monoxide in the blood. And yes, this is all over. This marathon. In the end, the speed has to be faster. But no worries, no worries. Okay, those who are listening, just tell me the answer. Honestly, over. Just tell me the answer. Tell me, the sportsman, which drug the sportsman abuse? What are the different drugs that the sportsmen misuse? Number one, yes, diuretics, good, diuretics. Number two, cannabinoids, very good. Cannabinoids. Number three, which painkiller? Opioid analgesics. Opioid analgesics. Yes, very good. Diuretics. Then opioid analgesics. Cannabinoids. And number four, steroids. Anabolic steroids. Anabolic steroids. These are the four drugs that the sportsmen misuse. Sportsmen misuse, right? One of them is called anabolic steroid. So, see, steroids are like the cortisol, the steroids. Anabolic steroids are testosterone, androgens. They will cause the anabolism, okay? Build up anabolic steroids. Now, they are the male sex hormone. But if males take them, see, unusually if you take them, they are causing more harm than good. And let me tell you what harm it can do. Right. So, why it is taken? It is taken for more muscle mass, more aggression, okay, more energy, more aggression, more muscle mass. But the opposite effects are there in a long-term use. And what are the opposite effects or the harmful effects? It will cause testicular size diminished, testicles diminished. Okay, so testes will diminish. Two things will become bigger. One thing will become smaller, testes. Two things will become bigger. Male breast, gynecomastia, prostate, prostatic enlargement, prostate, gynecomastia. Two things become bigger. One thing diminish, testicles, testes. Now testes will diminish though sperm production is also decreased sperm production. Why? Because already androgens are so high from outside, the body will think that already too much of androgen is there. Why we have to produce our androgen? Okay, and to produce the androgen, the hypothalamus is continuously sending that tropic hormones to the gonads, the gonadotropin. So hypothalamus and the pituitary, they can become silent. Well, no problem, you are already enough of androgens are there. So hypothalamus will not ask the pituitary, and pituitary also will not produce these gonadotropins, and therefore the gonads start decreasing. And these abnormal androgens, then they will cause development of acne, depression, anxiety, depression, acne. They are common in all, whether male or female, they are common in all. But in male, just remember what diminished and what can increase. In the females, what it can increase and what it can diminish. Okay. Now it can increase. It can cause the enlargement of clitoris. It can cause the enlargement of clitoris. Okay. In female, clitoral enlargement. Okay. Of course, due to that testicular reduction, infertility, sperm production ability decrease, no doubt about that. Sperm count decreases. So these are the male and female effects. Different male effect, different effects in females, different effect in males. There is a masculinization of female. The female become masculine, male-like, and gynecomastia. The male breast becomes female-like. The female-like mast in the man. The female-like mast. Mast means breast. Gynecomastia. And masculinization. That means becoming masculine. Now, becoming masculine is abnormal for whom? For female. For male, it is desired. For a desired matlab limits, not animal-like limits. Okay, and for female, it is of course abnormal. Of course, will come the voice changes, will come, hair will grow all over the body. Okay, bachcho, that's all about this marathon class. Already, like I take a promise, and you have been with me for such a long time, particularly these online students. Okay, I can see some names. Some names have come in middle till now. They are there, but some names are there from the very beginning. That is really good. Anish is one of them. Very good. And today exam is there. I pyondi beta. I pyondi. Now you have your food. Take some 10 minutes rest. Very important. 10 minutes rest. Okay. Close your eyes. Let your food get digested in your body. Let the food will heal you, heal your tissues, heal your mind. And then again, if you want, you can revise the question. And better, rather than reading, you can do the questions, and that will teach you more than reading the textbook. Practicing is more, because what comes, see, I prepared last 3 days, I prepared anything, like I made the time stamp बनाया था मैंने, इतने बजे से इतने बजे तक ये होगा, ये होगा. What happened? Whatever came by reflex, that happens. Because only for this day, if I keep on doing this marathon again and again for another 1 year, then it will come into my nerves. But since marathon is once in a year, right? So we have to prepare, and whatever we prepared extra, all gone. What is coming out? What is reflex? Body memory, internal memory, muscle memory. Okay, so develop that reflex system. All the best, bachcho. God bless you. Bye bye. कुछ भी नहीं है बेटा इस पे तो, nothing is there. Something we wrote there in that application. Okay, beta. God bless you. Bye bye.